You're the next speaker?
Yes, I am. I'm Josh.
I'm Ari Cohen.
Nice to meet you. Nice to meet you. I need to put on something, right?
Yes, you do.
I've never worn such a thing.
She doesn't work.
This time shows me 25 minutes. And then I have 5 minutes. See what happens. Oh, and
You go over 30 minutes.
give me fair warning before you start streaming. Yeah. Much more exciting to see the direct action.
Crocodile. אוקיי. אומרים לי מתי להתחיל או אני...
There's one thing I need to do actually.
wait. חכה רגע.
I need to set up the advancer, the slide advancer.
אוקיי. הוא צריך לארגן איזה משהו. תעזור לנו אולי?
גל, תראה המחשב שלו מחובר. שלא מחובר.
לא שמעתי לך. הוא רוצה את הקליקר להעביר למחשב אחר.
כן, תעביר לו משלנו אליו.
אז הוא הביא, הנה, סבא.
תראה שהקליקר על און.
אז כאילו מסמנים להתחיל או אני בטוח? Okay, welcome to the afternoon session. Our first speaker will be Joshua Plotkin from University of Pennsylvania. He uses mathematics and computation to study questions in evolutionary biology and ecology. Research in his group is concerned primarily with adaptation in populations. Related interests include the evolution of robustness and adaptability, the evolutionary ecology of viral populations, the nature of genetic drift, the dynamics of protein translation, and the evolution of cultural and social norms. And today he'll talk about evolutionary forces of cultural change.
Come on down. So thank you. I would like to start by thanking Zahi for having the vision, really, to conceive and organize this fascinating workshop. I will be talking about using tools from evolutionary theory, especially population genetics, to study cultural change, starting first with language and then turning to other aspects of human culture. So I will start with a few slides that you've already seen before, but they're so beautiful I'd like to show them to you again. The first is this terrific passage, this evocative passage from The Descent of Man, where Darwin says, "A struggle for life is constantly going on amongst the words and grammatical forms in each language. The better, the shorter, the easier are constantly gaining the upper hand, and they owe their success to their own inherent virtue." And as we know from Mark, Darwin here was not speaking for himself, he was really quoting the linguist Max Müller. Because Darwin saw the science of language evolution both as an inspiration for the development of a theory of organismal evolution, but also, and I think this is quite important, as a way to explain the radical idea of common descent to a skeptical public that could not understand it. We've also seen this slide of Schleicher's famous tree of Indo-Germanic languages, now Indo-European languages. Darwin was, of course, a frequent interlocutor of early linguists, especially Schleicher. And indeed, the fields of linguistics and evolutionary biology were born together, and they were fast friends from birth. Darwin was jealous, I bet, of Schleicher's tree because, unlike his own trees, which were just sketched in his notebooks here, Schleicher has names for these internal nodes. Because Schleicher, of course, had the record of the extinct languages in terms of text, and he had much more detailed information about these internal nodes than Darwin could have ever hoped to have had from the fossil record. Now, after this terrific birth of linguistics and evolutionary biology, where they were closely intercalated fields, I wish I could say that the subsequent centuries saw a continual cross-fertilization between these two fields. But that's false. In fact, there was relatively little subsequent exchange between these two fields. Until recently, we have seen in Quentin's talk and a little bit in Eskay's talk the use of sophisticated techniques from phylogenetics to infer the relationships amongst all languages. But by contrast to those sort of macroscopic questions about how different languages are related, contemporary linguistic theory about how languages change within a single community of speakers, within a single population, remains sort of a strictly Darwinian theory, fossilized, as it were, in the form that it was when Darwin and Schleicher were talking to one another. And so as a result, modern linguistic theory that tries to explain the forces responsible for language variation within a single community focus on natural selection, focus on selective mechanisms, Darwinian mechanisms, that may bias the language towards one direction or another. So here, for example, we have from Bill Labov, actually a linguist at my own University of Pennsylvania, in 3 large volumes on the principles of linguistic change within a single community, a whole dissection of forces, internal language factors, social factors, cognitive and cultural factors that may selectively bias a language's evolution towards one direction or another. What linguists have not developed, because they have not been in communication with evolutionary biologists through this previous century, are other ideas for other forces that might be at play in how languages evolve in a given population. And in particular, linguists have only recently appreciated that stochastic drift, which is so natural to a population geneticist, may also play a role in the transmission and evolution of a language within a community. Randomness in whom you happen to encounter as a learner may, of course, affect the fate and the variation of forms and sounds within a population. And so to take, for an example, a place where we might consider stochastic drift as, in addition to selection, as playing a role in language, I will take this classical example of English past tense forms. As we know, and this is where Pinker really cut his teeth, it is known classically that English simple past tense forms are sometimes irregular, but they tend to regularize over time. So Middle English help becomes Modern English helped. Middle English rot becomes Modern English reached, and so on. And linguists classically view this as the result of some sort of selective force that is driving language change. For reasons of economy or cognitive ease, it's just easier to remember the regular ed ending as the past tense form, and so as a result, that pressure, that cognitive pressure, forces language to regularize past tense verbs. But linguists have not really used, to much extent, actual population genetic methods to infer what really is the strength of this pressure, and are there any other forces that might be responsible for these types of language changes. And so our group turned to looking at large corpora that have been annotated of relatively short timescales. This is a corpus developed at BYU of 100,000 American English texts spanning about 150 years or so, comprising 400 million words that have been annotated to parts of speech. And so here we can finally look at the actual dynamics of 2 alternative linguistic forms. These are the past tense of the verb dive. So in around 1850, if you wanted to say the past tense of I dive, you would have said I dived, whereas today you're more likely to say I dove. And it's quite important that we have annotated text as opposed to, say, just Google Books, because when we read the word D-O-V-E, we know that this is the simple past tense of the verb to dive, as opposed to the white bird that flies around and brings peace to the world. And so with these corpora in hand, we can inspect in great detail, the way a population geneticist might, the frequency trajectories of alternative linguistic alleles. And so here, for example, I'm showing you, in fact, all 36 verbs that show any significant polymorphism in their simple past tense conjugation in these American English texts and their frequency trajectories from the early 19th century to today. And the question, of course, is what are the forces that are responsible for these changes in the given language community? And the two particular forces that we want to investigate are those of selection. There might be some force that drives one form to extinction over the other, but there could also be drift. And so we've developed ways to use actually fairly standard population genetic models of selection and drift that describe the probability of an allele being at any given frequency in terms of this advective term, where W here represents selection, some force that might promote one form over the other, but also include the possibility of neutral drift, that just by random chance, some individuals may learn from other individuals, and there might be random drift in the frequencies of forms, irrespective of any sort of direction. We've also developed a method to test, is there any selection at all? That is to say, a null hypothesis is that there's no selection at all and that everything is attributable to drift, compared to an alternative that W is non-zero. And when we develop and assign these tests and apply them to these linguistic data, we find the following results. On the one hand, shown here in color, there are several verbs for which there is definitive evidence of selection for one or the other regular or irregular variant of the simple past tense form. Nonetheless, the vast majority of the other verbs are completely consistent with neutral drift. There's really no statistical reason to imply that there's selection that was responsible for the changes in these frequencies. Just randomness in transmission is sufficient to explain the trajectories of the vast majority of verb forms. But the ones that are under selection are, of course, much more interesting. And so those are the ones I want to talk about for a little while here. Indeed, we find, for example, some examples of selection towards the regular form. So smelt, which we might have said in 1850, is now said as smelled, the past tense of the verb smell. And wove is slowly being replaced by the regular form weaved. Weaved is still in the minority, but we predict, of course, that it will eventually replace wove. The regular form seems to be selectively advantageous here. So there are examples of selective regularization, but there are, as it turns out, just as many, in fact, even more examples of selection for the irregular form. So sneaked is being replaced selectively by snuck. Dived is being replaced selectively by dove, and so on. So these results, which we can formalize, we can provide a p-value as to the fact that there really is selection for the irregular form, of course, contravene the standard linguistic expectation that verbs should regularize over time. So what is going on here? Why do we find selection for these irregular forms? Now, I can't really answer that question in any definitive way, but I can certainly share some interesting post hoc speculations about what is causing this pressure for the irregularization of certain verbs. And our main hypothesis has to do with rhyming. Changing frequencies of rhyming conjugations in the corpus as a whole may be responsible for a selection for irregular verb variants. And in particular, let's take this first example of dives being replaced by dove. This I attribute to the invention of cars in the 1920s. So cars were invented in the 1920s, and not all of you are native English speakers, but for those of you who aren't, drive is a strictly irregular verb in English. I drive, I drove. There's no drived. That word does not exist in English. And so cars were invented in 1920, or were common in 1920, and people started writing and speaking. I drive, I drove, I drive, I drove, I drive, I drove. And then finally they decided to think about diving. I dive, I dived? No, I dive, I dove, right? Just like I drive, I drove. In other words, the frequency of the verb drive, which has the past tense formulation drive, drove, increased rapidly with the invention of cars. And this exerted a pressure by phonetic analogy that selected for dove. Another way of putting it, of course, is that the invention of cars drove, dived, towards dove. Here's another example. Quitted, as a simple past tense, was quite common in 1825, but is now replaced by quit. I quit. And this, too, we believe, is driven by phonetic analogy. And there's a marked increase in the frequency of the it irregular conjugation in the corpus as a whole. And that is largely responsible. The main reason that is caused for that is because of the word split. So split's frequency in these texts increased massively around 1900. And the reason for that is because split acquired a new meaning in 1900. So split initially meant to divide in two. But then split's meaning split into two. And split means two things now. I can divide something in two, or I can say I split that joint, meaning I departed from that location. So split acquired a new meaning in 1900, and its overall frequency in the corpus massively increased. And so, of course, the rhyming conjugation, it increased, and this may have pulled quitted down to quit. And in fact, it's not just these two examples, but in all 11 out of 11 cases where the rhyming irregular simple past conjugation is on the rise among monomorphic verbs in the corpus, so too does the selection coefficient on a polymorphic verb form favor the rhyming irregular variant. And so in every case where there's some rhyming pattern changing in the corpus for whatever reason, that induces some form of selection for the irregular form of a past tense verb. Okay, so this is some discussion of selection on verb conjugation. I haven't discussed much about drift. I won't discuss much about drift except this one brief slide here. We can, of course, also infer the strength of stochastic drift that is responsible for language change, at least in these texts. And the most interesting and important result is that the overall frequency of a word in the corpus is strongly related to its inferred effective population size. So the words that are used more frequently have larger effective population sizes, meaning lower amounts of stochastic drift. So this has a lot of implications, because it means that a lot of the relationships between word frequency and word replacement rates of one form by another, which have typically been considered as selective effects, may in fact be explainable by purely stochastic effects, because the more frequent words will be less stochastic in their alternative formulations over time. So I want to also turn to discuss other types of morphosyntactic change. There are other aspects of morphosyntactic change that linguists were well aware of and have beautiful comparative evidence that these are changes that are driven by selection. And one beautiful example is due to Jespersen, who noticed a cycle of the form of verbal negation. So from pre-verbal negation, so in Old French we would have said je ne dis, to bipartite negation, je ne dis pas, to what is post-verbal negation, je dis pas. And je dis pas, you guys may know, is the sort of colloquial version of French that's spoken in Paris today, whereas you might have learned je ne dis pas if you actually took French in grade school. So there's overwhelming comparative evidence that this cycle of negation, of alternative forms of verbal negation, is due to selection, because we observe it, Jespersen observed it, in multiple, not just English and French, but in multiple languages simultaneously. So there's really no reason to doubt that there's some sort of selective reason that's driving these verbal negation patterns. The question is whether or not we can detect it from corpora of English alone, without any comparative evidence whatsoever. And so here, this is really just a positive control for our methods. We looked at about a millennium's worth of English texts from Middle to Modern English, summing over all verbs, and these are now completely parsed texts. We can ask, I'm using the word say here, but we can ask how often are different forms of verbal negation used over this millennia. And indeed, we can test whether or not there is any selection, and we reject the hypothesis there's no selection. So both of these transitions in Jespersen's cycle are indeed identifiable as being driven by some sort of selective force that's changing the way we negate words. Okay, now there's lots more to be done here in this domain of using population genetic tools to understand language evolution. And here I'll discuss some very preliminary work of using similar tools in the context of phonetic evolution. Phoneticists, and we'll hear more about this afternoon, I think, have a wonderful way of recording human speech, transcribing what was spoken. So this is someone who spoke the actual every millisecond of the recording is then assigned to what word and what phoneme is being uttered. And they can extract the so-called first and second formants of every vowel phoneme, which correspond to how high or low your tongue is in your mouth when you make the vowel, or how forward and back your tongue is in your mouth when you make the vowel. So this is a way of analyzing in a sort of a precise way how people form vowel sounds. What has been done by Bill Labov at University of Pennsylvania is to do this over time, which is an amazing task. It took his entire career to do this. He recorded on the order of 400 conversations of people speaking in African Americans speaking English, just colloquially, in Philadelphia, comprising the order of 2 million words, and these speakers ranged in time over about 120 years in their birth year. And so we have, and this is actually a figure from Bill Labov's early papers, the location of the first and second formant, where the tongue is when speaking each of these vowel phonemes, but also how those locations may change over the course of a century of spoken speech in Philadelphia. And here you can see, again, rich opportunity for us to begin to quantify and test hypotheses about, in this case, phonetic change in a single community of speakers. So, for example, we're beginning, this is just really work in progress, to infer the strength of drift, the strength of selection, stabilizing selection that makes you want to say the phoneme in a certain location, but also potentially the strength of directional selection that is moving the phoneme over time for some reason. So this is a famous vowel phoneme that has moved in Philadelphia over the course of 100 years. And there's lots of interesting, this is, of course, work to be published yet. There's lots of interesting, very natural hypotheses about why certain phonemes may be more constrained in their production and why other phonemes may be driven to change in their production over the course of 100 years, none of which I will talk about today. What I want to turn to instead is another cultural trait, baby names. This is a cultural trait that is sort of linguistic in a sense. What name you choose for your child. So to get us started, I'll show you here data on the frequency time series of the name Alice in the United States. So Alice used to be one of the most popular female names in the United States. Around 1880, it was one of the most popular names, but then it took a huge dive. No one really wanted to be Alice for a while. And then parents decided, okay, Alice is cool again, and we should start naming our girls Alice again. And so Alice is on the rise. So we have these types of time series data. This is, of course, a cultural trait that has shown marked variation over the course of this 150 years or so. And of course, we have the time series data for every birth, every first name in the United States over time. And there's wonderfully interesting dynamics here. And the question is, can we use population genetics to infer the forces responsible for these cultural changes? Instead of inferring a single fitness parameter W, as we did in the past, or earlier in this talk, what we really want to infer is how the fitness of a name depends upon its frequency recently. So we want to infer a fitness function, W of X. That is to say, as a function of the current frequency of the name in the population, how readily will a parent choose to use that name in naming their child today? And there must be some relationship between the frequencies of names in the population today and their fitness, their propensity to be copied by parents today. And I can tell you my intuition for this as a parent. When my wife and I were forced to choose the name of our firstborn child, I remember thinking strongly, well, we can't choose a name that's too rare. If we choose a name that's like unpronounceable, then the poor kid will have this terrible name and no one will be able to talk to him for his whole life. But at the same time, we don't want to choose a name like Bob or Joe that's so boring. Everyone, with apologies to any Bobs or Joes in the audience, we don't want to choose a name that is so common as to be boring. There's some sort of intermediate frequency. That's the name we want. And I'll tell you what name we chose in a little bit. But the question is, can we infer from these time series data what is the form of frequency dependence that explains the cultural evolution of these name uses? So that's a very hard problem because we want to infer an infinite dimensional object. And so the very first thing we do is make it at least piecewise constant, so there's a finite dimensional problem. And then we developed, my student Mitchell Newberry developed, what is essentially a piecewise constant expectation maximization algorithm. And in essence, he rediscovered a technique that has been used for a while in MRI imaging to infer the form of piecewise constant frequency-dependent selection that is most likely given the time series data on the changing frequencies of names in the United States. And this is the inference which we drew. And you can see this was quite gratifying to me as a parent. Indeed, we found that the names that are very rare, any name more rare than 1 in 10 to the 4.5 or so, those names have negative growth rates. These are the names that are really so rare that no one wants them. If names are super common, on the other hand, these are the Bobs and the Joes, names more common than, say, 1 in 1,000 individuals, these are too common to be interesting, so no one wants them either. They're sort of going downhill too. But these are the cool names here, right? These are the names that are like rare but not too rare. Our firstborn child, Linus, sits right here at 1 in 10 to the 4.5. I did this research long after naming our child, but I told him later how perfect his name was. He didn't believe me. But this inference, which has very tight confidence intervals, you can see, really explains a large proportion of the name data. So, for example, Alice in 1895 was almost the most common female name, but of course, as a result of that, it had a negative growth rate, and 100 years later, it had decreased in frequency quite a lot. So in 1995, Alice was now in this cool region where you could be a cool Alice, and so Alice went up in frequency, and now it's settling down to the only stable equilibrium on this curve, which is around 1 in 1,000. So these data are available across a whole range of cultures. You may be wondering, is this what happens in other countries? So this is what happens in other countries. We have named every birth and every first name in 6 different countries now, and we find this remarkable concordance of the form of frequency dependence across cultures. Of course, the actual names themselves in these countries are very different. So Silje, this is a name that ranges across the whole frequency domain in Norway, but it's vanishingly rare in the United States, as you might imagine. But nonetheless, the Norwegians have the same feeling about how to name their kids as the Americans. They don't want these really rare names, and they don't want these really common names. This is sort of somewhat universal form of frequency-dependent selection. By the way, just as a quick side note, Alberta, Canada, you'll notice every name in Alberta has a negative growth rate, and that reflects the fact that Alberta has a remarkably high immigration rate. And so, in fact, every name is going downhill in Alberta because immigrants, which are viewed as new alleles in our model or mutations, exceed the rate of replacement of any one name. So, in fact, we can actually look at the immigration rates in all these different regions and understand where the set point is on this y-axis. Okay, so hopefully I've been able to convince you that even across many different cultures, this very simple frequency-dependent model explains a large proportion of the variance in names. That's to say, even when the names differ between countries, the form of the actual name itself isn't what matters, it's sort of the frequency of the name that matters. And so this exchangeability assumption, which is at the core of our inference, is not a completely stupid assumption, is what I'm trying to argue. Except for cases where it is a stupid assumption. And here's one such case. Here is Tom Cruise introducing himself to Kelly McGillis in Top Gun 1987, and Tom Cruise says, "I'm Maverick." And Kelly says, "Maverick? Does your mother not like you or something?" And what's happening here is that in 1987, Maverick was a vanishingly rare name in the United States. No one was Maverick, and so Kelly thinks that Tom must have had parents who hated him to have named him Maverick. And you can imagine how we found this name Maverick. This name Maverick, its time series, well, let me show you its time series. You might guess what its time series is before I show it to you. So there is a little blip of Mavericks before 1987 because there was a US TV show that was broadcast in which the main character was named Maverick. But he had no effect compared to Tom Cruise. So then Top Gun is released in 1987, and really Mavericks start getting born after that. So right after Top Gun, there's something like 50 Maverick borns. The next few years, there's 100. Soon we have 150 or 200 Mavericks born. And so, of course, this is a name whose intrinsic allelic identity, Maverick, has some salience because of this cultural broadcast due to Top Gun. And so this is a name that we identified as being an outlier compared to the whole data set. It is the most unlikely name given the whole data set. So exchangeability is violated. In fact, we can find all of the Mavericks in the data set, all the names which are unlikely. So I don't really have much more to say. I want to stop here to leave time for questions, but I especially want to thank Mitchell Newberry, who's a terrific graduate student who did all of this work on names and on language. He's now assistant professor at University of Michigan. Armita Nur Mohammed, a postdoc who's now a professor at University of Washington, as well as tremendous and welcoming colleagues in the Penn Linguistics Department and funding sources. So thank you all for your attention.
We have a little bit of time for questions.
Thank you so much, Joshua. I would like to ask you about Sturtevant paradox. Edgar Sturtevant in 1947, I think, formulated a paradox according to which phonetic laws are regular, but they produce irregularity, whereas analogies are irregular, but they produce regularity. So this is kind of a paradox in linguistics, and my question is whether what you showed us, especially at the beginning of the lecture, weakens Sturtevant's paradox, because you believe that, say, if we dive, dove, drive, drove, etc. I mean, we have many, many verbs these days that have I-O, so there is a new regularity there, as opposed to the... does it weaken, or, I mean, do you add something that Sturtevant's paradox did not take into consideration because you look at cultural traits that are beyond analogies? And then if the answer is yes, my question is whether your lecture weakens totally what we heard, for example, from Quentin about the a family tree understanding of all languages. Because what you tell us is that family tree, if you look at it at the micro, is totally useless.
So as a neophyte to the field, I'm unfamiliar, in fact, with this paradox. But my understanding, at least from reading Pinker, is that despite the fact that I've is now a sort of fairly common conjugation, it's not near as common, of course, as the suffix ed. And so that's definitely the regular form. And it may be that changing frequencies of rhyming conjugations can sort of set small minority patterns that draw other polymorphic verbs or other polymorphic forms into that form, into that pattern. I don't think, however, that this work has any contradiction with the kind of research that Quentin was discussing in that entire field of phylogenetic research of the relationships between languages. There's this interesting divide, which I tried to describe at the beginning, between the macroscopic view of evolutionary theory applied to language, that's say the understanding of what languages are related to each other and when they diverged, and the microscopic view of what variation exists within a community of speakers. Now, on the one hand, it must be variation within a community of speakers that is in the end responsible for the divergences of languages, of course. But at the same time, the actual techniques to infer the relationships between extant languages are completely different and can well assume neutrality. I think safely assume neutrality, despite the fact that there may be selection within a language. So I don't think and in fact, this occurs even in biology, where we find lots of selection occurring within a population, and yet we're quite comfortable to use neutral models to explain divergences and the to-date divergences between species.
We have time for one quick question and even quicker answer.
You pointed out the cultural influence of moving pictures and automobiles. You could add television and so on. But there's, I think literature has an important influence, and especially among Hebrew speakers, where we have ancient texts that keep coming up and certainly form an important influence on the selection of words.
So I'm happy you asked that question, especially here in Israel. I didn't have time to say it, but now I do. I have a quick answer. In names, I would have thought that the biblical names, the John, the Isaac, the Marks, have like a much higher fitness than they should have given their frequency, because they have this incredible cultural salience. They come from the Bible, right? And to my surprise, I found that being a biblical name increased your fitness by only about a few % relative to what it should be given the frequency of the name. So this isn't quite words, but for names at least, having a biblical salience has a very small moderating effect on the fitness of the name. Whereas being named after Tom Cruise has an enormous effect. So the products of Hollywood have strongly outweighed the products of the Holy Land here. So thank you.
Thank our speaker. The next speaker comes from closer to home, Adi Stern from Tel Aviv University. Her research interests lie in the rapid evolution of viruses and their hosts. Viruses and hosts are at war. In order to survive, each side must constantly gain new weaponry. In her research lab, she uses a combination of computational biology, evolutionary theory, and experimental evolution in order to gain an understanding of this fascinating arms race. And maybe she will help us in our war against viruses in these troubled times. We will find in a moment when she talks about the evolution of competing cheater viruses.
Okay, done. Oh, yay. Okay, so again, I'd like to extend my thanks to Tzachi for this really inspiring conference. And so I study viruses, and most of my lab, we study virus genomics, virus genetics, and evolution. Where basically what we try and do is sort of decipher the language of viruses, right, that is embedded in the genetic code of viruses. But today I actually want to tell you a slightly different story, where what we try and do is crack something about the relationship between viruses, and so sort of the social evolution of viruses. And so I know everyone hears about viruses all day long now on the news, but I really want to take a step back and kind of define viruses before I begin my talk. And so what are viruses? And so viruses are kind of can be defined as the simplest organism on Earth, where there's a big philosophical debate if we can actually define viruses as alive or not, and I'm not going to get into that debate, but I'm just going to say that viruses are basically can be defined as a small piece of genetic material, which is either DNA or RNA, which is surrounded by a protein coat, which enables them to attach to a cell and enter it. And viruses infect every single form of life, whether these are humans, dogs, cows, fish, amoeba, and they even infect bacteria. And we're going to be talking about that type of virus today. So bacteriophages are viruses that infect bacteria. And the common ground for viruses is that they are parasites, so they are parasites of cells. Everything is made out of cells. Our own body is made out of cells. We have blood cells, we have heart cells, we have every tissue in our body is basically composed of cells. Everything that is alive is composed of cells, so bacteria are also cells, and that is why we have viruses that can infect bacteria. And so what do viruses do? Viruses hijack cells and turn cells into a factory that makes more and more viruses. And a basic scheme that describes how a virus replicates is that the virus, first of all, has to enter the cell, and that's why it has that protein coat that on the external that allows it to attach to the cell. Then it uncoats, and into the cell comes the DNA or the RNA, and it replicates. So it replicates, it creates more copies of its genomes, which are eventually then packed once again into a protein coat, and that's the last stage, exit, which is assembly and packaging. And we're going to get back to some of these stages later on. And so how do viruses evolve, and how does anything evolve? And so the major factor that allows evolution to occur are mutations, okay? And for viruses is in particular important because they evolve very, very quickly, right? And so one of the reasons I'm so interested in virus evolution is because viruses have basically the highest mutation rate on Earth. Okay, so what we see here on the y-axis is the mutation rate, which is the probability that a mutation will occur during a replication cycle. We're humans, we're down here. Our mutation rates are around 10 to the minus 10. Okay, that's a very, very small number. Whereas viruses, you can see clearly, both DNA viruses and RNA viruses have the highest mutation rate on Earth. And in my mind, the most interesting and exciting viruses are the RNA viruses, right? And so RNA is a form, a difficult, a slightly modified form of DNA. And you can see that they're the winners here in the race, right? They have the highest mutation rate out there. And these viruses include very well-known viruses such as Ebola, Zika, influenza, and the coronavirus that we're all aware of, okay? And coronaviruses are actually particularly interesting because they're right at the bottom here. They're actually not the fastest ones here. They're an outlier in RNA viruses because they have lower mutation rates, despite having slightly lower mutation rates than all the rest of these guys. As you know, they're able to create these very large and widespread epidemics. Okay, so that's why I think viruses are very interesting, both from an evolutionary perspective and from a medical perspective. And now let's ask what are the consequences of these mutations? And so this is going to be surprising, but most mutations are actually bad for viruses. Okay, and the analogy in evolution that is often made for this idea is, right, if you take a clock and you bang it with a hammer, are you going to get a clock that works better or worse, right? And so clearly the answer is worse, and that's the idea behind mutations, that if you take the genetic material of an organism and you change it, most chances are you're going to create something that operates less well, right, worse, and not better. And in viruses, this is in particularly extreme because their genomes are very, very, very small. Any mutation that you create, or most mutations that you create, are actually bad for the virus. Nevertheless, sometimes a mutation is neutral, which means it has no effect. They're sometimes also called silent mutations. And rarely, a mutation might be adaptive. And of course, that's what we're most scared of, right? Mutations that are adaptive to the virus means the virus that can grow better and can potentially, again, talking about human viruses, create epidemics. Okay, but going back to that notion of most mutations are bad for the virus, this is a concept that virologists are very familiar with. If you look at a viral population, and so, right, this is representing a cell, and you have all these viruses that just now exited the cell. Virologists know that most viruses that exit the cell are actually non-infectious. Okay, and they're non-infectious because of what I just told you before, because a lot of these viruses bear genomes that are mutated, and these mutated genomes are not able to complete a replication cycle. Okay, and so they're also called, the term is called a defective virus, a virus that is non-infectious. Okay, so most viral particles are defectious, non-infectious. Some of them are in particularly interesting. They're called defective interfering particles because not only are they defective, but they interfere with the intact virus, okay? So we can have this virus which is defective. It's not able to complete a replication cycle of its own, but it'll interfere with the so-called wild type or intact virus replication. And there's been a huge amount of interest in these defective interfering particles in the past years because the thought is, can we use these as antivirals? Okay, so in influenza, there's a lot of interest in people that are sick with influenza, and we can actually treat them with these defective interfering particles, interfere with the wild type influenza replication, and this will reduce the infectious load in the patient. And so there are huge grants now that are revolving around this use of defective interfering particles and clinical trials and so on. But I'm going to argue that we actually don't fully understand these defective interfering particles, what is the mechanism, how they interfere, and their effects on viral evolution. And today, our story is going to begin. So we're going to kind of try and answer that question that I just posed now. And to answer that, we looked at a model virus that we grow in the lab. And conveniently, it's a very, very safe model virus to grow in the lab because it's a virus that doesn't infect humans, it infects bacteria. And so the hero of our talk today is this virus called MS2. And so as I just mentioned, it infects the bacterium E. coli, which is a resident of our gut. It's one of the smallest known viruses on Earth, one of the smallest genomes on Earth, just about 3,500 bases long, okay, so 300,500 letters long. To compare our genomes, human genomes are 3 billion bases long. It's extremely fast evolving, and it's used as a model virus to understand evolution of RNA viruses. And so just to walk you through, this is the genome of the virus, just 4 genes, okay, that's tiny, it's a little bit of nothing. I'm always amazed how this really little bit of nothing is actually able to take over an entire cell, which is much more complex, and destroy it in something like 20 minutes. And so we have these 4 genes are the A protein. The A protein is what allows it to attach to the E. coli, to the bacterium. The coat is all this external part, the external part of the virus. The lysis protein is what basically lyses the E. coli cell, the bacterial cell, and allows virus to exit the cell. And the replicase is a very important gene. It's called a RNA-dependent RNA polymerase. It's what copies the genome, copies RNA genomes into another copy of RNA genome. Okay, and so we started with an experimental evolution setup where we evolve the virus in the lab, and this is something that we do a lot. We evolve viruses in the lab with the aim of understanding evolution in a very controlled setup that we can manipulate in the lab and see what happens, and we try and look at mutation rates and what's called the fitness landscape of the virus. And then we sequence all the time points so that we can understand evolution. And the first thing that we try to look for are actually those rare adaptive mutations that I mentioned a few slides ago, so mutations that are good for the virus. And mutations that are good for the virus should look like this. They increase rapidly in frequency over time, kind of like what Joss showed previously in a different context. Okay, and so these, we mark them immediately as mutations that look like adaptive mutations. And we were kind of happy because we see things repeating themselves. So we repeated the experiment twice in biological duplicates, and we saw the same colored mutations appearing here, and okay, so that seemed nice. But then we started looking into the nature of these mutations, and we were very, very surprised because the first one, marked in red, is delta 1764. It's a point deletion. Okay, so what is a point deletion? One base in the genome of the virus was deleted. Okay, so it looks like this. Okay, this U here is wiped out of the genome. Talking about a hammer, this is a big hammer. Okay, if you think about the genetic material, wiping out something doesn't sound like a good idea. And in evolution, we rarely see these kinds of events of one base being deleted. And so couldn't really understand how this is an adaptive mutation. And the second mutation that also you see in both of these is the orange one, A1664G. It's a silent mutation. It actually has no effect on the protein level. Once again, these are mutations that are normally not adaptive. And so this really doesn't make sense. And so we started looking into this even more. And so speaking of this deletion, we tried to infer what the effects would be. And so if the virus would have to chosen a mutation, this is probably the worst location for a deletion to occur. And so what happens here is that this gene, that I mentioned before, the replicase, which is responsible for replicating the genome, is basically becomes non-functional because of this point deletion. So this gene is wiped out. There's an overlapping gene here, which is called the lysis, which is responsible for lysing the host cell. And so what happens here is that the gene is cut into 2, so only the first half is created. But it turns out that the replicase, the form that is the different form of it's called frame shifting that is caused by the replicase, creates a second half of a lysis. So we get 2 halves of a lysis protein. And at this stage, we kind of wonder, do 2 halves of a gene equal a whole? Will 2 halves that are created separately function like a whole protein? And finally, there's also something which is called an RNA structure here, which is created. There's also another element here, which is called the TR loop structure. It's essential for viral packaging. So this bit of RNA that folds like this is what is responsible and what allows the virus to be packed, all right? So we mentioned that the virus, that's the third stage that I mentioned before. The virus has to exit the cell. It has to create its RNA. It has to be packed in a protein. This is what is responsible for it being packed. And in the structure, this last bit at the bottom here was, again, wiped out. That U is wiped out. And so this seems like a really, really bad mutation. And so what we hypothesized is going on is that this is what we call a cheater virus. Okay, so I'm going to define a cheater virus. And cheater viruses are actually these defective interfering particles that I mentioned before, but let's define them the way we look at them. So cheater viruses have two properties. First of all, they're defective, which means that they cannot infect cells on their own. They can infect a cell when they infect together with an intact so-called wild-type virus. Okay, so they can only infect if they infect together with a virus that provides whatever is missing. But not only are they able to infect together with wild type, but they have a selective advantage over wild type during infection, which means that more cheater progeny are created than the wild type progeny. Okay, and that's why they're defective interfering particles. And so we set out to prove this. And so first of all, what we did is we take the population of viruses that we have here, and this is called passage 15. And so we see there's a mix of all sorts of mutations that we see in this population. And then we dilute the virus. We dilute to the extent that only one virus can initiate an infection. This is called a plaque assay, one of the most critical property kind of assays that is performed on any virus that one studies. So this is what a plaque assay looks like. You have these plaques that are created just by one single virus. A plaque means that so the black part here, the background is bacteria growing or cells growing, and wherever you see a plaque, this dot, it means that the virus lysed, it blew up the cells there. And what we see here is that none of these colored mutations appear here. Okay, so there's only actually one mutation here that can create a plaque assay, which is 535G, which is not too important for this talk. But what this actually tells us is that this red mutant and this orange mutant, and actually all the other mutants that we see in the experiment, they're not able to create an infection on their own. Okay, so the conclusion is that almost all mutations in this experiment are defective or defective hitchhikers, which mean that they're together with a defective mutation on the same genome. And what we actually conclude is that there are two cheater lineages here, the red one and the orange ones, two major cheater lineages in our experiment. Then we went on to ask, what is the cheating mechanism of this deletion cheater? And glossing over a lot of work, we were initially stumped because, as I mentioned, that deletion, that cheater which has one base deleted, has three different defects. And so eventually we were able to prove that the defect is only because of the lack of the first one. So the lack of replicase is what is responsible for the defect. And so if we give the virus a replicase protein from the outside, it's able to replicate, and that means that these two halves of a protein are actually able to function. And the advantage is actually, turns out that what we thought might be a defect is an advantage. So the change in this RNA structure that is responsible for RNA packaging, for viral packaging, actually gives a virus an advantage. Okay, so what is going on here? This deletion cheater co-infects together with an intact virus. It steals its replicase protein, and then it's packed even better than the wild protein, so we get more cheater progeny because of that. The next stage that we performed here is we went into game theory, and so we wanted to model these dynamics using game theory. And so game theory is ideal here because game theory speaks about defectors and cooperators, where the cooperators are the normal intact viruses here, and the defectors are these cheater viruses. And so what we quickly realized is that what we should be getting here is something which is called snowdrift dynamics. And in snowdrift dynamics, we expect there to be a steady state reached, an equilibrium reached between the wild-type intact virus and the cheater virus. And the idea is that, so this cheater, which is the red, right, it should increase in frequency because it creates more progeny. It has an advantage over wild-type. But it can't reach fixation. It can't reach a frequency of 1. Because if it reaches a frequency of 1, that means that there are no wild-type viruses left, and then it has no one to cheat, right? It can't grow on its own. And so there's some steady state in the middle, so that's the model inference, the black line, and the red line is our experimental observations, and they kind of seem to fit the model. But we only performed the experiment until passage 15 or 15, 16, and we weren't kind of sure that this would continue, and so we said, okay, let's continue the experiment and see what happens. So is equilibrium maintained by more passaging? And this is what we get after continuing the experiment until 24, and I'd just like you to focus on that red line. Is it at equilibrium? The answer is no. This red mutation crashes, okay, over here, and it crashes over here too. And you see a bunch of other things going on, but mainly you see that orange mutation that I kind of partially ignored till that stage, going up, right? In both lines, the orange mutation is going up. And this was kind of we called it the demise of the deletion cheater and the rise of the silent or synonymous cheater. And at this stage, we were completely struck, and we had no idea what's going on here. And it led us to start examining this orange mutation more in depth. So silent cheater, who are you? And I'm going to really very briefly touch upon the mechanism of what we think this other cheater is doing. And so it's a silent mutation, which means it doesn't affect the translation of this here, of this coat protein. But what it does do is it blocks the creation of the lysis protein. This is called a ribosomal binding site. What it does is it prevents lysis from being generated. What this cheater does is that no to little lysis protein is created. So we get these tiny, 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 tiny plaques. More protein is created. So the virus is basically trapped inside the cell. The virus enters the cell on its own, creates everything, but it doesn't know how to blow up the cell, and so it's trapped. When it does co-infect together with an intact virus, the other virus, the attacked virus, creates the lysis, and then it can exit the cell. And it exits the cell very, very efficiently because, again, it creates more co-protein, and it's packed even better. And so we went back to the model. Okay, so now we understand more or less what this other cheater is doing, and what does the model tell us about this? And so we developed a model which is a three-way model. We have so the interaction between a wild-type virus and two cheater viruses. And the model very, very explicitly showed us that more or less the only possible explanation for this, right, this cheater going up and going down and the other one going up, is that the synonymous cheater, the silent cheater, cheats the deletion cheater, but not vice versa. Okay, so we have one cheater that is able to cheat the other cheater, and the deletion cheater doesn't know how to cheat the synonymous cheater. And the mechanistic explanation that we have here is that there's a mechanistic clash, right? The synonymous cheater creates a lot of coat protein. The deletion cheater binds this coat protein very, very efficiently, so it binds everything. And so it's packed before it gets replicated. And that would explain everything that we saw in this experiment. And so to summarize, what I've shown you here is that in our experiment, almost all the evolution, which we define evolution as change in mutation frequencies, changes in so-called allele frequencies, almost all the evolution in the experiment was actually driven by cheaters or conflict between cheaters, which I think is a very key point here. We think that cheaters are probably very prevalent in natural infections. We have other work that shows that we see these cheaters in human viruses. All types of human viruses seem to support these cheaters. They go up and they go down, and I think that they've been overlooked often. It's very hard to detect them without genomic tools and an understanding of their mechanism, which kind of raises questions as can we use them as therapeutic agents? So that red cheater initially would have been a very good therapeutic agent, right? Because it interferes with a wild-type virus replication, but as we saw, it goes up and then it goes down, and something else comes up instead of it. And finally, what I kind of really like here is that we see what I call the babushka or matrushka effect, right? We have viruses that are parasites of cells, and then we have cheaters that are parasites of viruses, and then we see a cheater of a cheater, which is a parasite of the original cheater, and I kind of wonder how long this kind of chain of parasitism can continue. And with that, I'll end. I'll just say that this was led by a tremendous group of talented graduate students, in particular led by Mohan Meir, Noam Daniel involved as well. And these are the collaborators and funding agencies. And thank you for your attention.
We have some time for questions.
Of course, the successful virus is one that lives in harmony with the host cell. So I don't know why it's defective from one point of view. It may be highly beneficial to the host-parasite interaction in the long-term survival.
That's an excellent point. Yeah, there's always some steady state that has to be reached, but eventually, if you think of it, it depends which entity is undergoing selection here. Is the population as a whole, or is it the population of the wild type and the population of the selfish cheater viruses? It depends which entity is selected, and of course, probably everything. Everything. They all interact, and I agree with you, yeah.
I have a question here, and
Oh, sorry.
it's a language question because of the conference. So do you think cheater is the right word? I mean, it has a negative connotation, but if you have such a small genome, and I mean, making it deleterious is the only way you can progress. So a cheater is a kind of way to explore the alternative pathways and then accelerate the adaptation of the wild type.
Again, I agree. I think it's a cheater in the local sense, that it reduces, right? It reduces the fitness of the population of viruses at that very point. But I very much agree, and one of the things that we're very interested in exploring is how this actually allows the virus to cross what's called fitness valleys, right? So let's say that red cheater, which is it's very, very defective on its own, but can it correct itself? All right, so if it can correct itself, then we get a virus which is a super virus because it's packed very, very well. And so yes, we were also thinking about these as different ways in which the population can eventually evolve.
So Adi, in these experiments, do you ever observe, or do you expect to observe a situation when these passages, the propagation of the cheater would result in the collapse of the entire system? Let us say because of the shortage of the replicates.
Yeah, well, specifically in the setup that we have here, since we perform plaque isolation, we ensure a supply of wild type all the time. We control the MOI, which is the ratio between the number of intact viruses and the number of cells. So in our experiments, we always ensure that there are enough wild type viruses to continue. I think I find it hard to see how the virus population would go extinct here. I think there would always be some either steady state or fluctuations in steady state. But it's something that's interesting. I'd be happy to discuss, yeah.
Well, if we can't beat the coronavirus, at least we can call it bad names. Let's thank our speaker. The next speaker is Elisha Moses, comes from even closer by, from right here, the Weizmann Institute. And for those of you who believe that physics is hard, he's working on biological and soft physics. And he will talk about organization of information in language.
Shalom. Welcome. First of all, thanks to Tzachi and to Wendy for organizing this wonderful conference, reminding us that science is fun and we can enjoy ourselves. And I'll try and describe what we're doing in my lab. It's a few stories concatenated together, a little bit of everything, and there's a lot of technical details in everything, but I'll try and stick with the underlying principles. And really the idea is that there's enough data available and the technology and the concepts have developed enough that you can really begin to quantify intuitive, simple questions about language, okay? It's not a very formal structure. It's not a deep building into the formal organization of language, but it's a simple structure where you can ask questions and maybe get answers. And I'll go over 3 simple stories. One is books, and ask you how memory is created in books. What's the role of correlation? Another is dictionaries, these things which build our language, and I'll show you that loops and self-reference are the center of what a dictionary is, maybe discuss a little bit about concepts in it, and finally talk about the toughest and maybe the hardest thing of all, which is how we actually talk to each other. This is not structured, this is not a book in which you can read things, but you actually have to go listen to people and understand how they're conveying their thoughts to each other. So what happens with a book? A book, you read the book, and somehow concepts are created a long time. So you have to look back and realize that language is a string of words. It works in time, one by one by one. Wendy Sandler made the point that in sign language, you have more directions. It's not quite linear because at one point of time, it's not a single word. There are maybe two words, two hands, two words, I don't know. But still, even if you have a vector, in one point of time, you're progressing word by word by word or symbol by symbol with time, and therefore, you have to take this mess, which is our thoughts, our internal structure, what we think of, and create that into a line, a single line. How does that occur? So in order to do that, the structure we look at is the text of a book. We define a graph in which two words that are close enough, which means they sit together within about 200 words, and we ask how many times they fall next to each other, and this defines a relation between all the words in the text, and we choose perhaps all the nouns, or we limit ourselves, but we build it so that you can actually look at the words and ask what comes out of this. And there are a few, you have to decide how long you take the text, and then what this window is, if it's 200. There are many details which lead you to defining a distance or some kind of relation between two words. And with this, you can build a matrix which is actually all the relations between the different words in this text, and then you analyze it somehow, and the singular value decomposition is one which defines directions in sort of word space. So you think of a direction in word space in which the reader is pointed at the moment, and the author takes him through concept space or word space into a trajectory like this. And if you look at the books, it's an eclectic selection of books, as you can see. And if you look at the books, you can see that the lengths vary and the number of words that you actually choose each time changes. And the whole structure has to be, let's say, flexible enough that you analyze the books in a way in which you can go from very simple books, let's say Moby Dick or Tom Sawyer, to things like Einstein, Naked Lunch, etc. The concepts that come out, or the major vectors, the directions in space, are very telling. Immediately you see in Moby Dick all the necessary words that you will need in order to build the topic of what you're talking of where you're going. You see both the whale and the boat, everything comes together. But then the fifth one is an interesting one because it deals with something very specific. So there's a concept here which we will get at, which is probably before they're leaving the harbor. There's a description which is very, very specific, and you cannot describe it with the other vectors. And so, with these in hand, we go back and we build a correlation of the words, and we ask if you take a word and ask how often it comes back again, how long does it take until you see it? And the form that we get, at least for this one book, in terms of different window sizes, you see that there's a line which is coming out which is very different from a random distribution. And that's already telling. That's telling you that over time, or in this case, the word distance, over time, if you're very close in time, then the probability of seeing this word is high, but it doesn't go down to zero even if you get very far in the words. And that is what we would call a power law in time for the decay of the correlation. And you can see that every different book has a different power law, so it's a different behavior, but they still have this decay of the correlation in time, which tells us that we're building memory in some way. If we go farther, we still use that word, bit less often, but we still use it. So the way to understand this is that what we, to recreate it, let's say, to make a model of it, we shuffle all the words and then build them again, but maintaining some kind of hierarchy. And think of this hierarchy as something which is like volumes, going to chapters, going to sections, etc., in which the book builds some kind of structure in which you are walking through. And this kind of shuffling maintains the power laws, or recreates the power laws, which you would get if you didn't have the shuffling before. So the conclusion is that by having this hierarchy that we built into the book, the way that we look at a book as having sort of a hierarchy which focuses us into a single concept, in this way we recreate or build up the memory. And that's something which is very measurable, very quantifiable. We can bring it out, and it is exactly how the brain uses a time, uses a concept, uses the ability to remember in order to build something which translates from language into the brain. The second topic I want to talk about is dictionaries. Dictionaries, very simple. This is our language, right? Now, you choose a dictionary, you choose a different language if you want. The different dictionaries are built by people, and what I will describe now depends on which dictionary you take. But the features that you see come up again and again in each of the dictionary. So what we do is we take a word, and then we begin to ask, how is it related to other words? Since the number of words is finite, you can take a word and look at its definition. For example, dictionary is defined as a book which explains or translates, usually in alphabetical order, the words of a language. So dictionary leads to these 4 nouns, and we have the beginning of a graph. Look at book, how it's defined, and continue in this way. And since it's a finite number, you can define them all, and you will have a network, a graph, which you can construct between all the different words in the dictionary. So if we had something which was memory before, here we're looking at the words themselves and asking how they are connected to each other and whether we can relate one to another in this way. Now, this has been done before. Dictionary graphs have been looked at before, and you look at this graph, and it has some properties. For example, the amount of nodes coming in has a power law, and there's a distribution around 4 of the number of words in a definition of a particular word. And this has been looked before, and something that these people noticed already in the 70s is that if you go over and over, if you iterate this graph again and again, so you take one word and you see where it goes and again where it goes, and these are the number of iterations starting from probably all of the words in a particular dictionary, then you get into a small subset, let's say from 100,000 words, you converge onto about 6,000 words, which are called the core, if you want. And these are the words which are actually used for definitions. All the rest of the words, this huge graph of 100,000 words, is not used in order to define. So there's a small subset, which is the core, the center of our language, which is what we use in general, and which is what will eventually converge or lead any word into. This is an example. So 6,300, this is the number if you take a particular WordNet definition of the words. But of course, WordNet is a bit complicated. It has a few meanings, and they all come together, and you can separate them. If you don't separate, if you just take the largest one of them, then you're left with something like 1,600 words. And if you compare them to other central definitions of language, which are, for example, there's the basic English by Ogden, who said you need no more than about 1,000 words in order to converse, and he defined them well. And there's a correlation between them, you see about 52%. And then if you take the minimal kanji words that you have to know by the end of high school in Japan, then you see that there's also an overlap, let's say, of 24%. And sorry, no, with the core, maybe 29%. And then if you just look at what words show up in general when you look at words from any text, then this would be, okay, something, 47%. No, sorry, 40%. So this is overlap between the core of the dictionary taken from WordNet, let's say, and central bodies of the English language or definitions of them. And you see that there's a high overlap, which says that the core actually is what you need in order to speak. So much for the core. Now let's look a little bit more into what goes on within the graph itself. And an interesting thing about the graph is that it has loops. Okay? It has larger loops, smaller loops. You can see different sizes of loops, 4 over here, 3 over here, 2 over here. So the graph that we've created has loops. And now the question is, what do these loops tell us? And we can count all the loops, right? It's everything. It's a database. Again, we have the tools of going in. These are questions you can ask, and here they are. So if it was just a random graph, this would be its shape, and it would average about 10 size of the loop, and that would be it. No, our graph is very different. There's a high number of very short loops, and there's a peak over here around 10 to the 15, which is funny. This is a funny structure. You don't quite understand it. And when we started to look into it, it turns out that this has to do with the fact that the words are not well enough disambiguated. So you have you have 2 loops in which they can connect, or 2 trajectories which can connect. And in this case, you can see that the word train has 2 meanings. It can relate to railway, it can relate to skill, to training, and this disambiguation, if you do it, then you can lower this peak, which is slightly an artifact. And what is really happening is that there are many loops which are smaller than 5, and this is a good example of the kind of loops that you are getting and which contribute. And now we want to say that the loops are central. The loops are very important. And why do we think that? Because when you build a language, you're at some point introducing new concepts. And it's very easy, or it's reasonable to think that when you introduce a concept, the graph must introduce a loop, because you have something new. If you had something before, like here, if you had something like, if you had something which is a tree-like, then this goes back in time. This is a new concept over here, and you can define it using the layers that you had previously, the time before. And you can build up and replace this new concept or this new word with concepts that you had before. On the other hand, if there's a loop coming in, and this is the new word which is defined by a closed circle, if you take any of these out, then you won't be able to define your new word. And in fact, these have also been revised so that they are now not, you cannot define them without using word B, let's say, and therefore the existence of a loop within the dictionary means that there was at some point a transition and you needed to be self-referent in order to define this new concept. We went to the history and asked how this actually coincides with what happens, and we looked at all the loops, and you give a histogram of their appearance, and it's the average time of all the words within the loop, and then you see that loops had a very high appearance sometime around Middle to Modern English, and then there's a new loop showing up when science and technology introduce new concepts which were not there before, and you had to suddenly revise the meaning of an atom or a nucleus. All these come in as new meanings, and you can see how this new loop comes in at a time when it's needed like this. Now, loops in general are important. You can see them everywhere. Self-reference, this is something that you cannot avoid. Most of the paradoxes that we know in the different in SETI or even Gödel theorem is related to some kind of self-reference, but that's probably going beyond what we have here. Again, as we said, within a loop, you have a probability to appear as a single unit, and this is the distance in time between two words within the same loop. And you can see that they often appear early, and if it were random, then their probability to appear together at this small distance in time would be much shorter. So in summary, loops are simple. They are sort of the basic unit that builds a dictionary graph. They make the core, which is all the basic words that we have. And we think that we have been able to show that the appearance of loops is related to the appearance of concepts. This means that the words within a loop will tend to appear simultaneously. And of course, we've been able to look at loops and define other loops with them. And that's also, again, within this single value decomposition, which gives you a description in sort of a generalized phase space. I will briefly go into the last topic. As I said, we've talked about memory, we've talked about self-reference. Both of these are sort of built into how we see language. Now we're moving into something much more complicated in terms of the noise or the analysis which you have to do, because it's inherently a human function. We do it all the time, and therefore humans are harder to analyze than books or the dictionary. So I'll be talking about prosody, and it is the music that accompanies the speech. It is an acoustic manifestation, so a physicist can easily look at it, and it encodes. First of all, we say something, which are the phones, but we also have a tone in which we say it, and there are many functions for the prosody. First of all, parsing. You know, the computer can extract the words that I speak, but it cannot say if I put a comma, a question mark, or perhaps a period. So the ability to actually say where we stop and why we stop, that's something, as Uri Alon showed, there's something to play with, which is the timing between the words, and the computer sort of misses that out. This is in the music. And of course, the fact that our emotional state is also conveyed by the tone, by the timbre, by the voice itself is quite clear. So some technicalities, we have to, there's a huge richness of the sound. You have to digitize it, typically at a high frequency, and then extract the phonemes. That's the words which we get. That's usually an automatic stage of text recognition. And then what we do is identify 3 variables which are slower and indicative of what is being said in the prosody. And that's the speech rate, the pitch, the intensity, and if you want the pitch and the sort of the formats go together. Now our UG is not a universal grammar. It's an ultimate goal, and it's to build a dictionary, at least for prosody, to have a list of all the possible prosodic utterances that we can do, and to construct sentences which are prosodic from that. This is how a typical manufactured...
We'll have some beer.
This is a manufactured...
Paul will have some beer.
Text which someone reads.
Paul will have some beer.
And we can take out the pitch. And that's how it sounds. Okay, so it's not too hard to reconstruct what we did before, and also to see that there might be an emphasis somewhere in there. The first thing we found was that in this utterance that we have, there's a segmentation, and that segmentation is not trivial. A sentence will be maybe 10, 15 words, but the way we speak is much more broken. We tend to spurt out something like 2 to 4 words, and we do it in an interval of about a second. And we think, or at least according to literature, Misha Tzodik's raised this, that it may be related to how much you can store in your brain. There's a short-term memory. You want to say something, you have to load it into the utterance mechanism. And what the kind of process that we envisage is that there's a small cassette being loaded with a program of how to spurt out these words, and you load a cassette, say the words, in the meantime you load another one, and this whole thing continues like this. So it's a feature of our interface, the interface between our software and our hardware. So here we have an example of this segmentation.
Well, I wish I was the person whose voice they used in the telephone when it tells you the number has been changed, and that I had to copyright and get some royalties. Like that lady, you know, you hear the number you have reached. They got a different woman.
So this was one long sentence, and you can see it's broken down. It's broken down to segments, and these are the things that we actually have to identify. We've been able to automate that. It's based on the fact that towards the end of a segment, there's a slowing down of the speech rate. That's very telling, and also there's a pause. And then if you have a sentence here, you can see that this is extracted automatically. You find those words where a new segment begins, and then you can begin to ask what is the different statistics that come out of this. And you can see, first of all, that the words themselves tend to be longer at the end, at the edges of these intonations. If usually a word is on the order of 300 milliseconds, it'll go up to about 400 or 500 here. And you can see that the phonemes also, if they're the last ones, then they elongate. So there's something about elongation towards the end of an intonation unit. There's something that there's a small pause also. You do combine those two and identify this together. I remind you that the computer at the moment cannot tell you if it's a comma or a period. It doesn't know at the moment. This is another example where there's a small error made, and you can see that the U, if you manually segment, then you can see that the U belongs here, but the U in this case
and it's
continues.
the assurance that your God loves you, that he seeks you, that he's with you, that he comes to you. It's this assurance.
Okay, now you notice this is real conversation. Okay, this is a real person speaking, and that's part of the problem in what we try to apply all the time. So a simple application of this is focus, is emphasis, and emphasis is used, just to assign different functions in speech. For example, if you have different questions, what do? I'm a doctor. Is there a doctor in the house? I'm a doctor. Okay, so you have to emphasize something. And then, are you a nurse? I'm a doctor. So there are different things that you can emphasize just by, and in this way signal what kind of question this is. And for example, if you can tell something about contrast, then this kind of emphasis is a contrastive one and tells you pick this alternative, not other ones. And this kind of emphasis we looked at into more depth, and that's something that is also...
That was scientific irresponsibility.
Okay, so here you can see...
That was scientific irresponsibility.
That you can identify very clearly an emphasis, but in the next example...
We didn't get around to moving to a real apartment.
No, so real is emphasized, but it's very hard to understand because it's not well recorded. Someone else is coughing. It's a mess. This is the level of quality that we have to eventually get if we want to solve in real life the kind of questions that we're asking. So what we did is we built a small data set in which you can see here 5 different utterances. This is the first word, which is emphasized, second word, and this is no word. So we have a few of these. We can learn these, and also the machine learns how to use them. And when we apply this to a full corpus, then you can see that we can reasonably well over the whole data set. We can both learn and afterwards extract the features of emphasis, and we've been able to take a huge data set, in this case the Santa Barbara corpus, and annotate a small subsample, a few hundred of these words, and get reasonable results, although you see they're very noisy, but when we apply them to the whole data set, we got very good results, and you can see that this is applicable, and you can actually extract the words reasonably well and find the emphasis automatically in this way. So I would say that, first of all, the emphasis here sort of keeps with this. This is the neutral declination, and you can see that the emphasis follows it, and we get very good precision and accuracy by using combination of both pitch and of intensity. Okay, conclusions. First of all, we've looked at memory, self-reference, and intonation/emphasis. Simple questions, but we can begin to get quantitative, reliable answers. Second, language is experimentally accessible even to a physicist due to the large database and the technologies that we have at hand. Simple approaches and the models do work. And now what we're trying to do, as I said, is build a dictionary for the prosody. I want to thank the team, especially Tirza, Tzvi Tlusti, who had great ideas, Jean-Pierre, who each one worked on one of the separate projects, and I thank you very much.
We have short time for questions.
Thank you so much. Elisha, is it? I would like to ask you about the second part of your lecture, the lexicon or dictionaries. I've noticed that you stopped at Ogden and some kanji, etc. There is Anna Wierzbicka who introduced natural semantic meta-language, in which she offers 65 primes only to define any word, any lexical item in any language. I asked her to define Jukurpa. Jukurpa is in Pidginjara or Walbri is dreamtime, or it's an aboriginal spiritual idea, and she defined it using 65 primes in 10 pages. My question to you is whether you believe that these kind of loopinesses, etc., have anything to do with our brain. In other words, do you really believe, because NSM is very attractive, this Wierzbicka's, which followed, of course, Leibniz. But my question is, do you really believe that when an Aboriginal person says Jukurpa, they actually have the use of all these primes within the brain, and whether we can use such loopiness as a description of our mental lexicon?
I will use Uri's good advice and rephrase this in, do you believe that loops are important to the brain? I think so. I think so. I think so. Not only for the structure of, language, of course, is an entry into the brain, and that's why we're studying it. But the importance of loops, I would say, goes beyond, and it's not quite clear to me how what we've seen encompasses what the importance of loops is going to be. I would say that self-reference, the identification of self, is built into the immune system. It's a necessary component of having a brain. The brain has to know what it is in order to look at the self and defend it defend its hardware. So every system which is slightly more complex has this self-referential part in it. The fact that we see it in dictionaries doesn't tell much about the importance, but I think it is part of something where loops will be important in general.
Time for a really quick question.
Hi, relates to the first part, and is, I wasn't sure I understood the relation to memory, and specifically how what you're doing is different from what is done with latent semantic analysis, which is a way to analyze a co-occurrence or distribution of words in text in general, and kind of derive meaning from that. So that's the specific comment. And the broader question is, there are quite a lot of quantitative approaches to the analysis of language over the past 20 years. And I'm was hoping you could say more about what novel insight you think this brings and how it builds on the existing work. And it's kind of a more general, broader comment. I think it's amazing. I'm a linguist and cognitive scientist, and it's wonderful to use tools and ideas from other domains to study language, but it's probably good if it's based on the existing knowledge that is already out there. And so I'm wondering if you can comment on what you think is novel in the approach that you're bringing.
This is going to be tough to take Uri's advice on. No. So the first thing is, what was the question? I would say that what we have shown in having power laws is that time decays in a very quantitative way, and that you cannot obtain these power laws without introducing some kind of structure. That's the only statement that we have. Absolutely, we know. However, we've never proved it. This is a quantitative way of proving that you must have structure. Without that, you don't get power laws. Now, as for the hubris of a physicist coming into physics, that's the second question, right? So this hubris has been noted before when physicists went into biology and they got shellacked, okay? And quite rightly, this is not the way to do it. However, the fact that you can quantitatively go into language at the moment, I think if you look at how Google or Amazon or these behemoths that go in, they're using quantitative analysis in full, and they are really using machine language. However, the role of the linguists there is minimal. So there, I would say that the concepts are less important than the actual product, than what you can get. And what we are hoping, or what we are suggesting, is to combine the two and use linguistic insight in a quantitative way. That's practically all that we're suggesting. So, linguists should indeed give the direction, like in physics, where the theoretician says this way, the experimental says, no, you're making a mistake, and they find out how to do it together. But I think that the data scientists and the linguists should somehow learn to talk. That's all.
As a linguist, I will give the direction to the poster session upstairs, and we will meet in 45 minutes. Let's thank our speaker.
Okay, so welcome to the last session of lectures. I remind you there's another session of playback theater in the evening after dinner. So our first speaker is Gilad Zuckerman. He comes from the University of Adelaide, where he studies the interaction and influences between languages, as well as the revival of languages, both the Hebrew language we speak, the modern Hebrew language we speak in Israel today, and the Aboriginal languages in Australia, which I believe is what he's going to be talking about today.
Thank you so much. Can you hear me? Dotan? Yes. Thank you so much. It's a pleasure to be here. Before I start, I would like to give this present to Tzachi Pilpel. Where is Tzachi? This is a special tie. Tzachi? Oh, you have to come to the stage. It's a special tie that I printed myself. You can see that my name is here. It's the Rosetta Stone in, as you know, hieroglyphs, Demotic Egyptian, and Greek. So please, thank you. So keep up the good work. And I would like to ask one big question today, and it has to do with language reclamation. Is it tree of life or network of life? The question is whether the Stammbaum or the family tree model is applicable wisely to language reclamation, which is the revival of a no longer spoken language? Or is it the case that the congruence principle is much better because it describes cross-fertilization between the revivalists' mother tongues and the language at stake that we are trying to reclaim? And the congruence principle might remind you of viral replication. We've heard a lecture about viruses early on. This talk is in support of cross-fertilization and natural selection as a perspicacious analysis of the universal mechanisms and constraints in language revival. But there are many related questions. For example, is language reclamation possible at all without cross-fertilization with the revivalist mother tongues? The answer is no. What can revivalistics tell us about language evolution and genesis? The answer is heaps or a lot. Heaps is Australian for a lot. And of course, the third question, which I'm not going to answer, but this is to be determined, can revivalistics tell us anything about evolution and genetics in general? And I think that this question is the most important question when it comes to Tzachi's idea with Wendy and with others about this beautiful conference that I have been enjoying immensely. So what is revivalistics? Revivalistics is a new transdisciplinary field of inquiry studying comparatively and systematically, on the one hand, the universal constraints and global mechanisms, and on the other hand, the particularistic peculiarities and cultural relativist idiosyncrasies apparent in linguistic reclamation, revitalization, and reinvigoration attempts across various sociological backgrounds all over the world. Note that I mentioned reclamation, revitalization, and reinvigoration. Reclamation has to do with a no longer spoken language, as in the case of Hebrew, as in the case of Bangla, that I will mention, as in the case of Ghana, and many other cases. Revitalization has to do with the revival of a severely endangered language, as in the case of Maori, Te Reo Maori in New Zealand, Aotearoa, the land of the long white cloud, also known as the land of the wrong white crowd by Maori people. And reinvigoration has to do with the revival of a language like Welsh, which has many speakers, Irish, but still it's endangered. As you can see, Revivalistics is a totally global enterprise. I paquet Corona. I travel all over the globe to revive languages or to assist in the revival of languages. This is in South Africa. This is in Namibia. This is in Thailand, etc. There are some slides here that I'm going to skip simply because I would like to get to the gist of my talk, so bear with me, I'm sorry about that. I thought if somebody did not come to the conference due to the coronavirus, maybe I will have more time, but I don't have time. So I'll just skip these. And I would like to ask this question. Tree of Life. This is the common Stammbaum model of the Afroasiatic family that includes, of course, Hebrew. Or Network of Life. This is my own hybridic model for the reclamation of Hebrew, resulting in Israeli. And you can see here that Israeli has many parents. It is in total, in diametric opposition to the Stammbaum model, in which every language has one parent. In the case of Israeli, which is my term for revived Hebrew, you have on the one hand a cuckoo, that's Yiddish, laying its eggs in the nest of another bird, tricking it to believe that it is its own bird. On the other hand, it's a phoenix. This is Hebrew, rising from the ashes. And on the other hand, it's a magpie, stealing from Russian, Polish, German, Arabic, Judeo-Spanish, etc. As you can see, it is not the result of reclamation tout court, but rather hybridization or cross-fertilization between various languages. I would like to talk about the congruence principle. The congruence principle, as opposed to the Stammbaum model, says the more contributing language a feature exists in, the more likely it is to persist in the emerging language, inadvertently, regardless of whether or not revivalists want that. Eliezer ben Yehuda, the main revivalist of Hebrew, might have hated his Mameloshen Yiddish, mother tongue Yiddish, but he could not help it. Yiddish survives beneath Israeli. Israeli, if you want, is not Retzach Yiddish, or the murder of Yiddish, but rather Yiddish Retzach, or Yiddish speaks itself, Retzach, beneath Israeli grammar. Let me give you some examples. Syllable structure. In Hebrew, which is the old language of Isaiah, not Isaiah Leibovich, another prophet, it was consonant, vowel, something consonant. So C stands for consonant, and V stands for vowel, and X stands for either consonant or vowel. In Yiddish, the syllable structure is sh, optional, so brackets means optional. Consonant, consonant, vowel, it's the only obligatory, necessary, compulsory component of the syllable structure in the syllable. Consonant, consonant, optional, sh. It also has, I have to admit, CC, a little bit like in Czech. You know, in Czech, strč, prs, kr, skrk, put the finger in the throat. I've just bumped into a Czech person, Heldman. And in Polish also you have komunizm. Nizm is one syllable. Therefore it's communism, otherwise it would be communism. But we know, because the stress is always, almost always penultimate, mil-el. We know that nizm is one syllable. In Israeli, surprise, the syllable structure is not the Hebrew syllable structure as you would have expected if you follow what the Academy of the Hebrew Language alleges that we speak the language of Isaiah, but rather it is identical to the main Yiddish syllable structure. So you have words like ktsitsa. Ktsitsa means meatball. If a Hebrew person would say ktsitsa, it would be kesiso. Kesiso. But we say ktsitsa, which contradicts the Hebrew syllable structure. Shrimps, we eat shrimps. Shrimps is in contradistinction to CVXC because you cannot have mps at the end, but in Israeli there is no problem whatsoever. Strudel, it's the @ sign, you know, like Gilad.Zuckerman strudel, you know, we use it as strudel. And of course, strungul, so there is no problem with the str in Israeli, whereas in Hebrew, it would have been esht run gul as istadion for stadion in Greek or stadium in English. So I can give you minimal pair, Platon, which means Plato. Platon is the Greek philosopher, but we cannot say Platon in Hebrew. We say Platon with a prosthetic a. But in Israeli, we say plastic. There is no problem whatsoever to say plastic. It's perfectly okay. In Hebrew, we would have said plastic. Another example is the minimal pairs, the following ones. In most contributing languages to Israeli, and unlike in Hebrew, f and p, as well as v and b, are phonemes rather than allophones. The result in Israeli are minimal pairs such as hishtavetz, had a heart attack, versus hishtabetz, was assigned. Needless to say, that minimal pair could have never existed in Hebrew, because in Hebrew, the v and the b were allophones of the very same phoneme rather than different phonemes. So it's a little bit like in Spanish, they say Barcelona, but Obama. So you cannot say Obama. The b becomes Obama, right? Barcelona is with a b, but Obama is with a w. The same with hitchaver, became friends, versus hitchaber, became linked. This is Israeli, minimal pair, impossible in Hebrew. Another example is leafer, to flick ash from a cigarette, versus leaper, to put makeup. And I've just made up this beautiful sentence, היום אני נותן לפחות 70% לפחות סטודנטים מאשר בעבר. היום אני נותן לפחות 70% לפחות סטודנטים מאשר בעבר. Today I give at least 70% to fewer students than in the past. Obviously, this was impossible in Hebrew, because in Hebrew, לפחות is the only way. You cannot say לפחות. In Hebrew, the f was an allophone of the phoneme p. In Israeli, f is an independent phoneme. Why? Because this is how it was in most contributing languages to the emerging tongue, which I call Israeli. Just as the syllable structure of Israeli reflects the syllable structure of most contributing languages to Israeli, the Stammbaum model is useless. In most contributing languages to Israeli, and unlike in Hebrew, there are no pharyngeal consonants. The result, Pake Victoria Hana from yesterday, pharyngeal consonants are pronounced in Hebrew, but not in Israeli. There is no, as a generalization, h, o, q, to, so in Israeli. And even when lovely Victoria said amok, she did not pronounce the q. Whereas in Arabic, if you tell somebody yakalbi instead of yakalbi, this is a horrific thing, because yakalbi means my dog instead of my heart. So a Hebrew speaker would say amok with a q. We do not have the pharyngealization in Israeli. As a generalization, Israelis do not pronounce this. In most contributing languages to Israeli, and unlike in Hebrew, there is no polarity of gender. The result in Israeli is esa shekel, which means 10 shekels, rather than asara, which is feminine 10, shkalim, which is masculine. So in Hebrew, there was an unbelievable phenomenon, not in Israeli, in which you put the numeral, which is masculine, together with the noun, which is feminine. This is why asara, with a a, which means feminine, just like in Italian, bella, right? It's a beautiful woman. If you go to a synagogue, and I guess that some of you have been to a synagogue, when the rabbi counts the people, he does not say chad, shtaim, shalosh, arba, because it is forbidden. There is a superstition that you're not allowed to count. He says, Hoshia et amecha huvarech et nachalatecha huraim unsem ad olam. So if he arrives in olam, it means you have 10 people on minyan, and therefore we can pray. I'm talking about male people. This is Judaism. Another example is phonosemantic matching. It's a camouflaged borrowing in which a foreign lexical item is matched with a phonetically and semantically similar pre-existing native word or root. So for example, if you look at a word like rakechet, which is rickets, it comes at the same time from rachitis, but also from rach, which means soft. So it's a disease of the soft bones. Sachar mecher comes from shacher macher, which is Yiddish for dark dealings. You can have it in German as schachern. I don't know if you have it in Austrian, Viennese, where is, you have it, schachern, like to haggle. And we say in Israeli, sachar mecher from sachar, which is trade, which happens to be the etymon, the ultimate etymon of shacher. And mecher, which has nothing to do with macher, which has to do with make in English. So macher limkor has to do with, it's more like kaufen or to sell. So this is a phonosemantic matching, two parents, one word. The same with mishkafayim, of course. Chaim Leib Chazan thought of skopeo, the Greek, which means look at, and matched it with a phonetically and semantically pre-existent root in Hebrew, shekepe, which allows the Academy of the Hebrew Language to claim that this is a 100% Hebrew term, because it has shekepe as its root. But actually, we know that this is only half of the truth. Dubbing, we say dibuv, but dibuv in the Bible means inducing someone to speak, whereas dibuv today is just like dubbing, which is, of course, etymologically unrelated to dbb. These are three, so buba, glida, this was in Google, I've found it, and it has, buba is a phonosemantic matching, which means doll, and ice cream, glida, which means ice cream. Kalking, in Israeli, we ask manishma, how are you? But literally it means what's heard. Of course, if you come from Yiddish-speaking family, you say manishma because of voserzich. But if you're, say, Nancy Brandes, he's a famous conductor, he would say manishma because of Romanian Chese Aude. He wouldn't say it because of voserzich. If you come from Polish, you would say manishma because of tsoswichach. If you come from Russian, because of Shtoslichna. And of course, if you are Georgian from Georgia, you will say manishma because of Ra'ismis. You have to understand that in German, you cannot say was hört sich with the meaning of wie geht es dir. Impossible. Kalking is very interesting when it comes to levels of Israeli or registers. We can say niyalo choshech ba'inayim, darkness has been made in his eyes, which comes from sizim gevo finster in the Eugen in Yiddish, but then sizim gevo finster in the Eugen comes from chashchu enav in Hebrew, which results in chashchu enav, which means he heard bad news, and niyalo choshech ba'inayim, which means he heard bad news, but actually it's a different kind of register. Ve'al kol mitnagdenu anachnu metzaftzefim is the last sentence in the anthem of the battalion for the defense of Hebrew. And what is interesting here is that they say, we do not give a damn on all our opponents, which means the Yiddish speakers, but they use a calque from the Yiddish within the same anthem inadvertently. It's shocking. Because meletzafzef, which means to whistle on somebody, comes from Yiddish, mir pfeifen sich auf unsere Säunim ois, etc. Okay, I would like to mention some Aboriginal parallels. I am a language revivalist, so I do not only do linguistics, I also go to the field and work with Aboriginal people about reviving their tongue. So in a way, I evolve in the active sense of doing evolution, so to speak. These are the Bangala people who participated in the first Bangala workshop that I conducted. This is 2018. You can see that there are more and more people joining the reclamation of the linguicided Bangala language. I should not bother you with details, but out of 330 Aboriginal languages, only 13 are alive and kicking, so 4%. 96% were killed. When I say were killed, I mean it. They were killed. People were not allowed to speak these languages, so there is an ethical reason to do that, not only a utilitarian and not only an aesthetic reason. So calking in reclaimed Bangala, the word for computer is Gabiwa. It comes from Gaga Bibi Waribirga. Gaga Bibi means brain, Gaga means head, and Bibi means egg. So Gaga Bibi means the egg of the head, which means the brain, so the egg inside the brain, Gaga Bibi, and Waribirga means lightning. So it's kind of electricity, so this means computer, because you have to understand that in 1844, there were no terms for electricity, no terms for computer, and we use a dictionary from 1844. Bangala Gabiwa is modeled upon Tereo Maori Rorohiko, which means lightning brain, but also computer. Roro means brain and Hiko means lightning. And of course, also, if you know Chinese, Tian Nao, deriving from Tian, which means lightning, and Nao, which means brain. So of course here, if you only look at lexical statistics, you might think, oh, Gabiwa, Gaga Bibi Waribiga, this is a Bangala word. But actually, what we see here, and this is the beautiful camouflaged horizontal gene transfer, we see Chinese and Maori surviving beneath Bangala. So if somebody comes in 100 or 1,000 years and says, oh, look, this was a very Bangala Pamanyungan language, actually, they totally overlook the fact that we looked at Chinese. This is my criticism of the Stammbaum model when it comes to lexical statistics. Look at the horizontal gene transfer here. Ruthlessly rootless verbs, verb templates. Binyanim in Israeli. Hishpritz means to splash. It has no root. It has a stem. The stem is spritz, as in Yiddish. Hiklik, it means to click on the mouth, for example, to do this. And this is from click. There is no root. There is only a stem, just like spricht, sprechen, sprechen, sprach, gesprochen, spruch, I-E-A-O-U, the upload in German, or sing, sang, sang, song in English. Of course, we have fikes, which means to focus, as opposed to fixes, which means to fax. So you can see a minimal pair. Of course, we cannot say fikes for fax, because we would have destroyed the prosodic structure of fax. If we say fikes, there is a vowel between the k and the s, whereas in fax, there is no vowel. Facsimile, yeah? Fibrek from fabrica, piprek from paprika, and of course, le narrative, to produce a narrative. I heard Professor Igal Schwartz from Ben Gurion University say le narrative, which totally contradicts the verb patterns in Israeli and in Hebrew. Israeli neologization is something that happens a lot. Maflutz magnuv, it's a new adjective pattern. You put the, it does not exist in Arabic. Of course, in Arabic you have majnoon, madrub, mahabul, etc., but not maflutz and magnuv. For avoiding risque translations, I'm not going to translate it. The speciation is more gradual than one might have thought. There is vertical gene transfer even after the alleged speciation, see, e.g. Homo sapiens sapiens, that is several percent Homo neanderthalis. We are all Neanderthals, as you know, to a certain extent. My wife has more Neanderthal genes than me. I would like to say that I only have several minutes before I have to end my talk, unfortunately, so I will just mention the founder principle. The revivalists' mother tongues are primary contributors to the emerging language, even if the revivalists do not want them to be. The word order of Israeli is subject, verb, object. It is not the same as Biblical Hebrew, which was verb, subject, object. It is much more, it is Yiddish, subject, verb, object, and most revivalists' mother tongues. The word order, for example, in Vedic Sanskrit was free due to the presence of 8 cases. In Hindi, most revivalist mother tongue of Sanskrit, it's subject, object, verb, and surprise, in Neo-Sanskrit, as you would have expected if you know revivalistics, it's subject, object, verb. The same with Ghana, the Aboriginal language of Adelaide. Subject, object, verb, like most Aboriginal tongues. This is the old way. In English, it's subject, verb, object, and in reclaimed Ghana, it's subject, verb, object. In other words, when you look at the syntax of the emerging tongue, it reflects most of the contributing languages, not the language you're trying to reclaim. Hawaii is another language that people are trying to reclaim. There is ho'e ho'e as opposed to ho'i ho'i, but both of them are pronounced as hoi hoi, so you lose this minimal pair of the original because of American English, because in American English there is no distinction if you want to say ho'e ho'e and ho'i ho'i. The founder principle says that Yiddish is a primary contributor to Israeli because it was the mother tongue of the vast majority of revivalists in the crucial period. And I would like to say that had the Moroccan Jews arrived in Israel first, had Ben Yehuda, born Perelman, been an Abarjil or Abarjel, Israeli would have been a totally, but I mean totally different language, much more Semitic. I would like to say that the case of Israeli and other revival languages, this is how we should call these languages, revival languages, demonstrates that the reality of linguistic genesis is far more complex than a simple family tree system allows. It might be the case that each language has a single parent in the normal course of linguistic evolution. I'm not convinced about that, but this is not the topic of the lecture today, but definitely not in the case of revival languages. Thank you so much.
So questions, anyone?
Is there a question?
Yeah, hi.
Oh, sorry.
Yeah.
What's your name?
Alon. Alon.
Alon, yeah.
Yeah, thanks for the talk. What do you think about the idea of analyzing Israeli, the Israeli language, as a Creole language, with taking the structure of Yiddish and the lexicon of Hebrew, as has been proposed?
Thank you, Alon. It's a wonderful question. The question is whether Israeli is a Creole. The answer is absolutely not. And the reason is very simple. If you look at Creolistics, the main difference between Creoles and non-Creoles is a sociological difference. In the case of Creoles, you have a society of employers or enslavers and slaves or people who work in the plantations. The people who work in the plantations are trying to speak the language of the employers or the colonizers, but they do not know, they do not have the knowledge to acquire it properly, in quotation marks. The case of Israeli is the diametric opposite case. Eliezer ben Yehuda knew Hebrew extremely well, much better than we Israelis know Hebrew today. The point is that sociologically, it's like the literati, you know, the ones who know the language very well, are trying to teach the language to the kids. But of course, in language reclamation, it's the kids who determine the grammar of the language, not Eliezer ben Yehuda and Bialik, etc. So sociologically speaking, the two scenarios are diametric opposites.
Thank you very much. What do you say about the possibility that some of the phenomena you described could be due to other factors besides influence from the other languages? So, for example, other Semitic languages have lost pharyngals, and if I'm not mistaken, spoken Arabic is subject, verb, object, whereas classical Arabic is verb, subject, object. Now, would you attribute those changes to influence from other languages, or are there other natural historical changes in language that can explain them?
I would say very clearly that you should never follow a red herring by finding a Semitic language that serendipitously shows a similar phenomenon of course, one, as opposed to a plethora of phenomena, and therefore kind of use it in order to weaken the hybridic genesis. Let me remind you of two things. Firstly, if the reclamation of ancient Hebrew were successful, I would have not expected to find all these failures in quotation marks, because I would have expected to speak a language which is as the language that Eliezer ben Yehuda and others wanted to reclaim. So we would have surpassed 2,500 years of evolution, of organic evolution. This is one thing, as opposed to Arabic and Aramaic, that were, in the case of Aramaic, Kurdified, in the case of Arabic, influenced by foreign languages. There is no possibility that you can argue that Hebrew evolved and evolved and evolved, because in 135 AD it died as a spoken language. And a language that is not a mother tongue, you cannot even talk about, So there was a guy called Goldenberg, and he argued, of course wrongly, that Israeli, my term, is a result of evolution of Hebrew. So for example, in some languages you have f and p, they just become different phonemes, just like in, I don't know, or ch and h, he just says, well, the h lost its pharyngeality. What do you mean? The fact that we do not pronounce the ch has little to do with organic evolution. It has to do that the speakers of Israeli were Yiddish speakers, so they could not pronounce the ch. I can pronounce the ch, but when I speak Arabic, it's not my mother tongue. You've heard so many accents in this conference. Why do you hear so many accents? Because people cannot get rid of their original tongue. And this is exactly what happened in Israeli. So whom should I believe? Goldenberg, that says that there was divergence and convergence? It's shocking that a linguist can argue such a thing. Whereas I know historically that no person spoke Hebrew as a mamelosh and as a mother tongue in 1000 AD. Pake Haramati, there were some crazy people who believed that Hebrew never died. Even Chomsky's father, Zev William Chomsky, not Chomsky, Chomsky, he believed that Hebrew never died. But I think that this is palpable poppycock. So no, it's a red herring. Thank you, Wendy. I hope I'm not too blunt, but I'm Israeli.
How do you explain the fact that the very first generation of Hebrew speakers, what you call Israeli, couldn't speak Yiddish? The very first people who were born in Israel and spoke Hebrew as their mother tongue could not understand or speak Yiddish.
You are talking about the people who were born in Israel in when?
At the beginning of the century, let's say the 1920s.
I hope you understand that the vast majority of revivalists of Hebrew spoke Yiddish as their mother tongue. The fact that their children did not speak Yiddish has little to do with what I'm trying to say, because the point is that the parents of the first speakers of Israeli were Yiddish speakers. The children were exposed to Yiddish whether they wanted it or not. The Yiddish that survives beneath Israeli is not because they studied Yiddish. They acquired, they did not learn the language from the parents.
They did not speak, they did not understand Yiddish.
So what? But this is exactly the point. I want to make it very clear. Language acquisition is not learning. It is something that happens automatically. I've heard the term learning about first language acquisition. In linguistics, we do not use learning. This is horrible to use learning. This is acquiring, acquisition. When you acquire a language, for example, I want now to reclaim Bangla, or I want to reclaim Latin, and I teach you Latin, inadvertently, your Latin will be Israeli-sized by my mother tongue. You do not, even if you do not know Israeli, let's say you are American, but I teach you my Latin, you'll be, of course, influenced by American. But if you're a child who is taken to a family of Latin speakers, and you do not know American, you do not know Israeli, you will not help it. You cannot help it. So you don't need to know Yiddish in order to speak Yiddish within Israeli. Of course not.
Okay, thank you. We don't have time for any more questions. So the next speaker is Tzachi Pilpel. who, as you all know, is one of the organizers of this meeting. And I'm glad I have this opportunity to thank him. So Tzachi, thank you for... I think what I've heard most people say outside when I asked about this meeting is that it's beautiful. I think
Pleasure.
It's a very good compliment. So Tzachi's lab uses experimental evolution to study dynamics and the mechanics of processes that underlie evolution.
I have the medulla. Yes. Okay, hello everyone. It couldn't be as emotional, I'm sorry. Good, so I'm in my lab we study real evolution, biological evolution, but as you know, I have a passion for science, so let me try to adapt what I'm discussing today in analogy to language, something that I would like to call genetic onomatopoeia. If it's not really onomatopoeia, don't take it too seriously. This is a question for the non-Hebrew speakers among you. One of those objects in Hebrew or in Israeli is called kos, and the other is called bakbuk. Can you guess which is which? I don't need to be Victoria Khana to pronounce bakbuk to give you a huge hint that this is probably the bakbuk and this is the kos, right? But if I ask you one of these objects is called kiseh and the other shulchan, in principle, you shouldn't be able to tell me which is which. Right? There's nothing table-ish in a table, there's nothing chairish in a chair. In principle, you cannot know. And if we use the term of signified and signifier, the signified is the object, and the signifier is the name that is given to it. Excuse me for using lousy linguistic terms. We know from de Saussure that the connection between them, in the majority of the cases, onomatopoeias are just exceptions, beautiful, but exceptions, the connection between them is totally arbitrary. Namely, we could have called kiseh shulchan and shulchan kiseh. We could have agreed that a chair is a table and a table is a chair, and everything would work. It wouldn't make much sense to call this bakbuk and this kos, but otherwise, in the majority of the words in any language, onomatopoeias are rare, and the arbitrariness of assignment within the signified and the signifier is the norm rather than the exception. Okay? What about the genetic code? Can I use this analogy just to convey to you something that we have discovered in the lab? So in biology, at the heart of genetics, we also have two systems of chemicals that I would like to refer to one of them as the signifier and the other one as the signified, and this is the heart of the genetic code. So let me teach you about the genetic code in a few minutes. So the DNA and the RNA, you heard about RNA from Adi, the viruses that have RNAs, their genomes. The DNA, the RNA is a language written over an alphabet of just 4 characters. These are the famous A, C, G, and T. In RNA, it's U instead of a T. It doesn't make much of a difference. It encodes for amino acids, which come in an alphabet of 20, and since 4 cannot encode uniquely 20, there is a system of codons, triplets of nucleotides, that uniquely define each amino acid. So, for example, this amino acid up there is consisting of a sequence of 3 uracil nucleotides, etc. And as you can see immediately, it seems that the genetic code obeys the arbitrariness because there is no simple connection between the chemistry of the nucleotides and the chemistry of the amino acid encoded. In fact, in this example, this bulky amino acid, this big amino acid, is encoded by small nucleotides, and this small amino acid is encoded by large nucleotides, suggesting that at least in terms of the bulkiness, there is a totally arbitrary assignment between the codons and the amino acids, between the nucleotides and the amino acids. And the structure of the genetic code conveys that notion fully. There is an arbitrary assignment, as long as it's agreed upon in organisms, that conveys or that maps triplets of nucleotides into amino acids.
Let me tell you a little bit about the structure of the genetic code. How do we read it? Again, it's triplets that encode for amino acids. So for example, the first amino acid, phenylalanine, is encoded with a U at the first position, U at the second position, and U at the third position, although C at the third position would also encode for that same amino acid. So in that respect, UUU and UUC are synonymous, but the code is arbitrary in the sense that, in principle, any combination could have encoded for any amino acid. In that respect, it reminds me of the Morse code that only has 2 types of characters, a dot and a dash, as opposed to 4 in DNA and RNA, and the Morse code is arbitrary in the same sense, because if you look at E that only has a dot, there's nothing dotty or dottish or dotty whatever in an E. It was just agreed that this is how you encode for E. And the same goes for the rest. This is not to say that this code or these codes are random, not at all. The genetic code is, we believe, is a result of very profound evolutionary selection. It's an excellent code, much better than many arbitrary codes, as much as the Morse code is an excellent one. For example, the most frequent signs in the language are characterized by short encoding, and similar characters have similar meanings, or mathematical quantities have similar encoding. So it's an excellent code in that respect. And the same goes for the genetic code, but it's arbitrary in the sense that there is no absolute connection between the signify and the signified. Now, let us take this knowledge to tell you something that we have recently discovered in the lab. This is an artistic view of a cell. Cells don't really look like that, but this is just to convey the notion that cells are a very complex entity. They are very crowded, and on top of that, molecules appear to have addresses, destinations within the cell. And Orna Choder is here in the audience. She was actually the first to have brought that extreme order to my attention, to the attention of many others. Molecules in cells have addresses. They don't just float within the cells. How does the cell know how to destine each molecule within the cell? There must be a language, a zip code, in fact, that would be embedded within the proteins and the mRNAs that would tell the cell where to put each one of them. Let me give you a more simplistic view of the structure of a cell, and this is a cell here that has a DNA, it has RNA, which is a transcript that goes to the cytoplasm where it's translated into proteins, and I want you to refer in particular to 3 types of proteins that cell makes. There's the proteins that are inside the cell, there are proteins that are secreted from cells. They are going into the medium. If it's a unicellular organism, they help build communities, public good, or weapons. And there are proteins that reside within the membranes of cells. So 3 types of cells. And the question is, how does the cell know the zip code for each of them? And for many years, the zip code was thought, and still is, known to reside at the level of the protein itself. There is information in the protein that doesn't only specify the function of the protein, but specifies where within the cell it should be located. But we thought, could there be some sense of genetic onomatopoeia that would allow the cell, already before translation of the protein, where there is only RNA, would allow the cell to say, this RNA is, when translated, it's going to be a protein that is secreted outside, and that RNA, when translated, is going to be a protein that is embedded in the membrane, and the third RNA, when translated, is actually going to reside within the cell. So if that were possible, it would have amounted to some onomatopoeia structure, perhaps resembling the bakbuk more than the table, but the question is there any reason to believe that there is such a code, that there is any such onomatopoeic structure? So for the biologists among you, you could of course ask me immediately, what are you talking about? We know about the signal peptides. Signal peptides are sequences embedded in the translated amino acid that tells a protein if it needs to be translated and if it needs to be, or if it needs to be embedded in the membrane. But we know that the signal peptide model is incomplete in the sense that many mRNAs that encode for secreted and for membranal proteins, they find their destination close to the membrane before they have been translated. And their translation, even in situ translation, does not necessarily depend always on that machinery that recognizes the amino acid sequence. So we asked ourselves, is there a code that perhaps resembles a genetic onomatopoeia? It would be embedded at the mRNA. It would allow the cell to tell ahead of time the destination of a protein. So let me tell you about one important property of amino acids, and that's the property, a chemical property called hydrophobicity. Hydrophobicity, as you know, is the tendency to hate water, and in fact, since the membranes of cells are made of lipids, if you hate water, you love membrane. If you love water, you hate membranes, and vice versa. So one can measure for the 20 amino acids this tendency of them to hate water or to love a membrane, and you can sort them. These are the amino acids that are hydrophobic. They like to be within the membrane. These are the hydrophilic. They don't like to be the membrane. And let me superimpose now the genetic code on top, and suggest to you, and this is partially based on an early observation that was made by Eitan Bibi and Chaim Prilewski here at the Weizmann. Some of it is actually known to people from even before that. An interesting observation, if you look at the genetic code and you color the amino acids based on this chemical property, whether they like or hate water, you see something that is hard to ignore, and this is that the first half of the left half, of the genetic table is mainly characterized by hydrophobic amino acids. And the right one is mainly characterized by hydrophilic amino acids. That means that there is perhaps, if I may use this analogy, a non-random assignment between the chemistry of the signified and the signifier. Perhaps to the extent that, and let me just mention that what characterizes the first two columns here, the U and the C, is that they belong to a family of nucleotides that are called pyrimidines, whose nickname is Y, as opposed to A and G, who are purines, whose short name is R. So this means that a codon that encodes for a hydrophobic amino acid takes the form of NYN, N being any nucleotide, because you can be wherever you want in the first and in the first base. Y specifies that it needs to be either U or C, which is this combination, and then it's followed by any other nucleotide. So if you want to encode for hydrophobic amino acid, usually a Y in the middle would ensure that, usually. Now, remember also another property, and it's important for the rest of this journey, that the code is redundant in the sense that if you have specified the first and second nucleotide within a codon, the third is typically free to mutate. Namely, synonymous codons would be different in the third position of the codon, and they would still encode for the same amino acid. So these are two important properties of codons. Hydrophobic amino acid would be NYN, and the third position is going to be relatively flexible in evolution. So the question is, could cells use that regularity, that interesting non-random assignment between the chemical properties of the codons and the amino acids to generate a zip coding system that would not be dependent on translation of the protein? So let me tell you something about membrane proteins. This is a segment of a membrane of a cell, of a hypothetical cell. And as you see, there are many proteins that traverse the membrane. And if you look at this particular one, it has typically 20 amino acids in a row. This is what it takes to traverse the membrane. And since the membrane is hydrophobic, it's made of fatty acids, you need hydrophobic amino acids in order to reside that. It's like, oil likes to be with oil, never with water. So we realized that a sequence, the nucleotide sequence, not the amino acid sequence, the nucleotide sequence of a segment that goes through the membrane should look like many repetitions, perhaps 20, of the form NYN, 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 etc., perhaps uninterrupted. Okay? So could that serve as a way to destine the mRNA for a membrane protein to the vicinity of the membrane before it was even translated? If so, that could function as a zip code that resides at the RNA, and it predicts ahead of time the result of translation without the need to translate in reality. Okay, but then we thought, what about secreted proteins? Secreted proteins, on one hand, they don't have hydrophobic amino acids. On the other hand, their mRNAs would better be translated in the vicinity of the membrane. That would facilitate their secretion to the external milieu of the cell. So there is a problem. On one hand, it would have been nice if they had this pattern. On the other hand, there's no reason for them to have the pattern because they don't have hydrophobic amino acids. But then we realized if we just ignore all the spaces in this long sentence and rebuild it again, putting the spaces with one shift to the right or to the left, we are getting something that secreted proteins could in principle use as their zip code at the mRNA level, despite the fact that they are not made of hydrophobic amino acid. And why is it? If they have codons of the NNY form rather than the NYN form, from the point of view of an entity, hypothetical at the moment, that reads the text without awareness to the gaps, it looks just the same. Because what's common to both of them is that you have pyrimidines, this Y, in jumps of 3. So we said, perhaps originally a system has evolved that would take that NYN repetitions as a zip code to localize mRNAs for membrane proteins, but secreted proteins could have co-opted that system and use the repetitions of Y in jumps of 3, but with an offset such that utilizing, in fact, the redundancy of the genetic code. Because if you remember, I told you that on the third position of the codon, you can essentially put almost always any nucleotide that you want. And if a Y is needed, this is exactly the place to stack it, evolutionarily speaking. So we've examined that. First, bioinformatically, and when I say we, I mean in collaboration with Jeff Gerstlab in our department, with Cohen Zontag as the first author, and indeed it turns out to be the case. Indeed, it turns out that membrane proteins feature uninterrupted runs of NYN, 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 whereas secreted proteins tend to feature runs that are also uninterrupted of pyrimidines of Ys in jumps of 3, but in an offset of 1 nucleotide. In fact, when you do the statistics, this is exactly what you are getting. I'm not getting into the details, but I'll just tell you that we have now a new regulatory motif that is present. It's a word that tends to repeat itself within the mRNAs that encode both for membranal proteins and for secreted proteins, and it has several interesting features. For example, it indeed features uninterrupted runs, 10, 15, sometimes even more, of pyrimidines in jumps of 3. This is very highly unusual. At random, with proper randomizations, etc., you wouldn't get it as much as you see it in reality in secreted proteins. It has additional interesting properties for the biologist among you. It can appear in the UTRs. As long as you have it, this is a localization signal, we believe, and indeed we have shown that increasing or decreasing the content of these pyrimidines in the mRNAs of proteins that are encoding for secreted proteins can have an effect on their production and secretion, etc. So now I want to tell you towards the end how we utilize this interesting motif that could not have been, by the way, discovered by the regular motif finding algorithms. It's a very elusive motif otherwise. It was only through the inspection of the genetic code, perhaps this analogy to the onomatopoeias that allow it to be discovered. But now we want to take it to answer a very important question in evolution of cooperation. And a lot of what I know about evolution of cooperation is from Martin Nowak, who gave a beautiful talk in Ilanit. I hope many of you heard it last week about the difficulty to evolve reliable cooperation. Why is it difficult to evolve cooperation? Because the tendency, as Adi Stern showed us in the previous session, the tendency to, or the temptation to defect is always there. And we are looking at a particular cooperation. So this motif that I told you about is present in many of the public good proteins that yeast cells and other cells are producing as a way to form their communities. And one such protein is called SAC2. It's present in a yeast that we love to study. And what SAC2 is a secreted protein. It's symbolized here with this X here. And what it does is that it cleaves a source of food that is otherwise indigestible. So now imagine a community of yeast cells. They live together and they secrete that protein as a public good. This is a very communal thing to do. Each of the participants secretes something, and then you have a functional society. Everyone contributes the effort, and everyone can enjoy that. We want to ask the question, to what extent this motif that we discovered that tells the cell to what extent to secrete or to donate some of its resources to the community, to what extent this is actually governed genetically by the motif that we have discovered. And this reminds us, of course, of the classical prisoner's dilemma, in which the temptation to defect upon others is very large. And in fact, many of the challenges in understanding how evolution has emerged is to understand how it was encoded genetically in such a stable way within biological systems. So what we thought was that perhaps the syntax of this motif encodes a semantics that governs the decision within such a iterated, perhaps prisoner's dilemma game, whether to cooperate or not. Now that we have a knob rather than a switch, defect or cooperate, a simple-minded dichotomous option, perhaps evolution could use that by adapting the structure of this motif, the syntax of this motif, in order to determine what would be an evolutionarily stable strategy of secretion versus retention for each of the public good proteins that a cell does. And I think I will not have as much time as I wanted for questions if I go too deeply into this. So let me just say very briefly that what we do, and when I say we, it's my two very talented students, Ruti and Dvir, who are, I think, here in the audience. What we do is that we actually engineer yeast cells and change the syntax, and we hope also the semantics of these motifs for this particular gene that encodes for a public good protein, and determine whether they would secrete more or less, how generous would they be, but again, not in a dichotomous fashion, but as an analog knob. And we co-culture now different variants of this motif in order to perhaps get insights into the evolution of this motif. How did it evolve over time? There's much to say about it, and even much more Dvir and Ruti tells me from result from just a few days ago, but I think I will rush quickly to the questions. And before that, just the acknowledgements. These are the people who have contributed. I will not read their names. And just at the very end, towards your questions, I want to show you what did I find about questions in evolution. And I couldn't resist that after Josh's talk. Oh, where is it? Disappeared. Let me find it. So this resource that Josh and Martin and many other people are using is really marvelous. If you go to Ngram Viewer, you can type in any word, and you would see how frequently it was used in many different languages. So Einstein, Frankenstein, Sherlock Holmes, etc. At some point, after playing a lot with that, I wanted to see what happened to punctuation marks. And in particular, I looked at the question and exclamation mark, and I had the intuition to pick up German, and this is what I saw. And this is just to motivate the question session here. So this is the usage of question mark and exclamation mark in German literature over the last 200 years. And we see a trend that is tantalizing, I think. We start 200 years with questions being 2 times more frequent than exclamations. Then the gap gradually narrows down. Alice would have been much more popular than question mark in these times. And it closes, and just around the First World War, and then it opens again, but it closes around the Second World War, and then it reopens again. By the way, the same trend, very similar trend, is observed in Italian. No phylogenetic, although historic relationship is obvious. It is not observed in English, in which the gap has safely been constant. So I think this is interesting as a way, perhaps, to understand evolution of even consciousness of people, and also to motivate you to ask me questions. So thank you very much.
So questions?
Yeah. The genetic table?
Yeah, where the Y wouldn't be a good predictor.
That's right.
So I just wondered if these proteins, when they're made, start going in the wrong direction.
Yeah, that's an interesting question. So the question was, in the table that I showed you, there was some interesting correlation between the property of the nucleotide and the property of the amino acid, but indeed you have noticed, Tamar, right? Tamar noticed that the correspondence was incomplete, imperfect. Indeed, it's imperfect, and this is indeed when you try to find statistical rules within biological data, it always comes with imperfections. So I don't know if it's an imperfection that we ignore and cells ignore, maybe we ignore and cells don't ignore, maybe it destines wrongly the mRNA to the wrong destination as a result of that. I think it's a good question. Maybe, yeah. Maybe if you have something with an exclamation mark, that could be used too. Which one? Hebrew. You want to see Hebrew? It's amazing. You'll be shocked. Israeli, whatever. That's Hebrew. Two things to be noticed. First, there is this bump here. It actually feels like an artifact to me. I can't understand it. But I checked when do they cross, and that was in 73. Okay, so wars in the history of nations have a weird tendency to change relationships between question marks. English, yes. I knew it. All the questions are going to be on that and not on motif. No, you have to ask me a question about the regulatory motif, and then I'll show you English. Yeah, over there. I'll show you English in a minute. Yeah, you.
Okay. Do you have a candidate to read the signal?
Oh, good. Who reads the signal? I told you that we think, at least metaphorically or idealistically, not metaphorically, we think about a protein that would recognize that signal. It should be a protein that doesn't care, for the biologists among you, doesn't care about the reading frame, because as we said, the Y can be in any offset, as long as that's what we think. As long as this is in jumps of 3 nucleotides, it's only the ribosome that knows how to identify the reading frame. The rest of the entities in the cell, even in the cytoplasm, are not known to have that, except for some interesting exceptions in splicing, perhaps. So, but it's a long way to say that we don't know. But in Jeff Gerst's lab, they are trying now to identify this protein. So what they do is that they put within a cell something that has this very strong pyrimidine content, and something that has exactly the opposite, the R content, the purine content. And you pull down the proteins that are binding each one of them, then you send them to mass spectrometry to identify the chemical nature of the binding proteins, and there are some interesting hints. That's what I can tell you at the moment. Moti. Do I have time for more questions?
Yes, but please repeat the question.
I will repeat the question. The previous question was who binds the motif?
You showed the last slide very quickly, but what I understood is that cells that cannot secrete SAC2 cannot compete with their... Have I understood it correctly? And the question is why?
Yeah, no, you understood it correctly. I just went way too fast. So the story that unfolds from this very small sample that we did, now we are doing it on a much larger scale of many, many alternative motifs. Each cell is getting a different version, a different policy of what to produce and what to secrete, and they are forming a community of various degrees of cheating. This was a simple experiment in which you either a very strong cooperator, a wild type, or a very weak cooperator. When we grow them alone, the cheater, of course, doesn't succeed, especially when we simulate what would be the result of their growing together, but in a naive simulation that does not take into account the cooperation and donation among them, it is predicted that the cheater would die out. But when we actually grow them alone, and that was the other slide that I didn't show, the cheater is actually doing very well. It is Together, yeah. When we actually grow them together, a co-culture of the three strains together, the cheater is actually quite happy. It is sustained in the population, presumably because it benefits from others. Whether something similar to what Adi showed us, that it could be later on disappearing from the population, or perhaps lead, due to the tragedy of the commons, to the demise of the entire population. We are actually looking at it now, but it doesn't seem that the population has collapsed.
So according to this, it's better to be cheater. I mean, you don't need to
Yeah.
have the energy to secrete and you...
Yeah, I hope, if one is allowed to hope what to expect from an experiment, I hope it would not be the result. We did some theoretical predictions, some mathematical modeling that suggested that perhaps when there is a whole continuum of cooperation, not just a binary switch between cooperate or not, perhaps there are some regimes of the space of parameters that allow coexistence of the various versions, but indeed we'll see that very, very soon. And towards the very end, I'll just show you how English looks like. Thank you very much.
So the last speaker for this session is Eugene Koonin. He comes to us from the National Center for Biotechnology Information, and he uses analysis of genomic sequences to reconstruct major events in the trajectory of evolution that make up the story of life on Earth.
One minute.
No. Right here. Good afternoon. It's a great pleasure and an honor to be here. The first thing that I have to say, of course, is to thank Tzachi and the rest of the organizers for inviting me to speak at this unique, wonderful, exciting meeting. And also perhaps to thank them for putting me in the last slot. I take it as a sign of great trust. It is the result of my using far too general title, which somehow fittingly got cut off here. It was evolution of genomes and evolution of languages, tantalizing analogies and major differences. I will try to tell you something about that. But in reality, of course, I will be far more specific and try to talk about this grammar of protein domains and somewhat beyond. So, this is my lab at the National Center of Biotechnology Information on the NIH campus in Bethesda, Maryland, very near Washington. And we study evolutionary genomics, various aspects of genome evolution, theoretically and more empirically. We study genomes, we do not study languages most of the time. So I feel like a bit of an imposter here, but I'll try. But it's hardly possible when you deal with genomes not to think about them as languages. So not to think about languages of genomes and genomes as languages. So, nearly 20 years ago, like probably a few other people in this room, I had the honor to participate in this momentous project, the Human Genome Project. And here are all our faces. You cannot see them very clearly, but each of you is somewhere here on the cover of Nature. And immediately, of course, primarily popular literature, but not only, started to talk about the genome as the book of life and started to talk about the language of the genes. It's inevitable. So here are some nice, in my opinion, quotes from a paper published as a Nature Insight review shortly after that by David Searles, an influential bioinformatician, on those sides, the language of the genes. So he says that linguistic metaphors have been woven into the fabric of molecular biology since its inception, and actually, as we have seen long before, pretty much since the inception of evolutionary biology in the modern sense. And he asks the question that we all have to ask. Do these analogies go deeper? And if so, can the methods developed for analyzing languages be applied to molecular biology? Further interweaving of these fields will be instrumental, he thought, in extending our understanding of the language of life. And this is what we are going to explore a little bit during the next 20 minutes or so. So we shall talk about protein domains at this point. And domains are structural, functional, and evolutionary units of proteins. So proteins consist of one or more domains. So a multi-domain protein looks this. We can treat a gene or protein or its product protein as a sentence like this, and each domain can be treated as a bird, as goes in this quote from Alice in Wonderland, "Curiosier in Curiosier," cried Alice. So these domains are all different, and these are identical. It's common for domains to be repeated in proteins, just it's relatively common for birds to be repeated in a sentence. So there are many multi-domain proteins encoded by genes of all organisms, especially those of highly complex organisms, such as animals, such as ourselves. And at some point around the Human Genome Affair, my colleagues and I dubbed that phenomenon domain accretion. With the increase in organismal complexity in the course of evolution, multi-domain proteins, the same, so to speak, protein, orthologous proteins, as we call them, tend to accumulate additional domains, providing perhaps for additional complexity, as we can see on these schemes for some rather beautiful examples, in my opinion. So domain accretion parallels, and perhaps to some extent underlies the increase in biological complexity. As you can roughly see in this slide we have here, the distribution of the number of domains and proteins in yeast, Worms elegans, and fly Drosophila melanogaster, and you can see that there the fraction of multi-domain proteins with many domains is much greater in the worm and especially in the fly compared to this relatively simple single-celled fungus. The tail of this distribution, so to speak, is much heavier. We'll see more of these distributions very shortly. Also, I would like to note that in the course of evolution of complex molecular systems and in the course of domain accretion, there are notable semantic shifts and acquisition of new meanings. As I illustrate here in this schematic that shows the evolution of the key proteins involved in RNA interference, a eukaryote-specific system of defense against infectious agents, viruses, and others. So here's this protein called dicer, which has been assembled from multiple domains that occur in different prokaryotes of this helicase domain of archaeal origin and these ribonuclease domains of bacterial origin. Then they're combined together. They acquire a new, they undergo a semantic shift and acquire a new function in the biogenesis of small interfering RNAs, which was not characteristic of any of the constituent domains. So, so much for the introduction, and in the main part of my talk today, now I want to address this question. How similar are words and domains in terms of the statistical properties of the distributions and combinations. So here is this distribution that has been seen already in a few slides in this meeting, and it could not have been otherwise because it's very, very familiar. It's a power law distribution where you have the rank order of frequency and you have this domain occurrence, the frequency itself, or counts of domains and counts of words in a fairly well known and relatively short text. The similarity of these distributions is quite striking. Actually, this is known in linguistics as Zipf's law, a power law distribution like this, the exponent gamma approximately 1. And this is not terribly surprising because this seems to be the general feature of evolving systems. You can see this distribution with a known degree of the internet, of a variety of other networks, biological, social, and otherwise, anywhere pretty much where we have an evolving system of interacting entities. It seems to reflect what Zipf called the principle of least effort, and E.T. Jaynes called the maximum entropy principle. So let's say in the human genome, the domain frequencies follow a power law, which in linear coordinates looks like this, not very informative, and looks much better in double logarithmic coordinates as more or less a straight line with this tail with the slope of roughly -1, which is already interesting a little bit because not only qualitatively but quantitatively it's very similar to what is seen in the corpuses of written in natural languages. So what we are going to deal with over the next few minutes is the n-gram analysis that was so beautifully illustrated in Tzachi's talk. So bigrams, doublets of words, doublets of domains, like this. We have these bigrams in this protein and these bigrams in this protein, and we can do all kinds of analysis of these bigrams. What we immediately can observe is that the increase of biological complexity, we see a steady increase in both the number of domain types and especially in the number of bigram types, reflecting the increasing complexity of biological organization, most especially in animals. So, again, not very surprisingly, the domain bigram distribution, just like the distribution of the frequencies of individual domains, follows a power law, but with a different slope around 2. And clearly, the lower the slope, the greater the complexity of the organization, the heavier the tail of rare combinations. And so here is the census of these slopes of power law distributions for domain bigrams in roughly increasing order of biological complexity. We deal here with eukaryotes only, and you can just see that these slopes are greater than 1 going up to 3 in unicellular eukaryotes, indicating the abrupt drop and the relatively small number of combinations, and a much gentler slope in plants and especially animals, indicating the greater number of combinations, reflecting greater and perhaps underlying greater complexity. So what we have seen so far is a very notable statistical similarity between the distributions of domains in genomes or in proteomes and the distributions of words in natural languages. For unigrams, individual domains or individual words, we have Poirot distributions with a slope of roughly -1. They're, again, quantitatively close. And for bigrams or combinations of two words, two domains, we have seen the slopes around -2, again, highly similar. So domains, for short, or more specifically, the statistical properties of domain distributions are mathematically similar to words. So far, so good. But I said that I was going to talk about grammar, so I will now go in that direction. So, very simply, just taking this from Wikipedia, we have heard very good discussion of this issue in Martin Nowak's talk at this meeting. Basically, grammar is a set of structural rules governing the composition of clauses, phrases, and words in a natural language. So I was rather struck to find out that the idea of a conserved grammar, of what we may call universal grammar, in a sense simpler than that of Chomsky, which was explained again by Martin in his talk, goes back at least as far as the 13th century to Roger Bacon and his book Grammatica Graeca. Grammar is one and the same, wrote Bacon, translated into modern English, of course. Grammar is one and the same in all languages, substantially, though it may vary accidentally in each of them. Very interesting proposition, and I would like to explore that proposition in terms of the grammar of domains in proteomes of different life forms. So, using language models or n-gram models, what we can and should explore is the probabilistic association of words of domains. In other words, what is the probability of a domain, let's say this red one, given the preceding one? And if we use what's called generative grammars, we can, given a language model, construct an artificial random genome that follows some general rules of grammar, but not others, to be compared with real genomes. The key notion here, as in many, many other areas, is entropy. Basically, we can have disordered soup of domains, and then it can be organized to some extent into distinct combinations, acquiring a degree of order. So an interesting question here is, what is the entropy loss in this ordering, which is obviously equivalent to information gain. This has been explored in detail for natural languages, and the universal entropy of word ordering across linguistic families has been recorded. And it has been shown that roughly 3.6 bits of information is gained through formal grammar in any natural language. So the bigram language models go like this. They give maximum likelihood estimates of the domain number n, given the occurrence of domain n minus 1, like this. This estimate converges at the ratio, the counts of this bigram to the counts of this first domain in it. So, how much information does the main language gain by following the grammar? Let's explore this and compare to natural languages. So the entropy of a language model is calculated like this, so this should remind those who know, and I hope that's the majority, of the Shannon formula, and the entropy of a bigram model is calculated like this, which is basically a very similar formula that uses, instead of the count of domains, the count of bigrams, and the logarithm of the ratio of the count of the bigram to the count of the first domain. And then we will use 3 forms of relative entropy. One is the basic form unigram to bigram, how much information we gain by moving from unigrams to bigrams. Then difference between unigrams and randomly shuffled bigrams, and finally shuffled bigrams and the observed bigrams. So we can construct these plots. The number of domain types, the count of domain types, and the entropy of unigrams and bigrams. And we see that in prokaryotes and bacteria and archaea, both unigram and bigram entropies increase with the number of domains, so the diversity increases. Interestingly, in eukaryotes, the number of the entropy of unigrams increases, the entropy of bigrams counterintuitively perhaps goes down, probably reflected proliferation of domain families, the high prevalence of repetitive domains in eukaryotic proteins. Generally, though, entropy of natural languages increases with vocabulary size, and so does the entropy of proteins. And here is the key thing that I want to show today, I believe, more or less universal relative entropy gain. These graphs show distributions of absolute entropies of unigram, bigram, and shuffled bigram in eukaryotes, archaea, and bacteria. And more importantly, these are these 3 types of relative entropies. And let's just pay attention primarily to this relative entropy, the difference between the entropy of bigrams and unigrams, and we shall see that these values are very close within, show sharp distributions, narrow distributions within each of the domains, with the lowest in archaea, slightly higher in bacteria, and slightly higher in eukaryotes. A quasi-universal entropy, relative entropy, quasi-universal information gain, as can be seen more clearly here. This broken red line shows these quasi-universal values with remarkable anomaly in animals, in metazoa, which possess the highest complexity and show the highest relative entropy, highest information gain from the grammar of the domain language, whereas archaea show the lowest. So basically, these are the conclusions that I can make from this, that there is a universal near-constant entropy of protein domain text, so to speak, a quasi-universal grammar of protein domain languages. Sensu Bacon, not necessarily sensu Chomsky. Both domain texts and natural languages possess universal grammar in that sense, or quasi-universal grammar, but domain language is much simpler. The information gain is only about 1.2 bits compared to about 3.6 bits in universal languages. Now, after all, we as the humanity managed to invent something more complex than the language of our genomes, which may not be unexpected given the high prevalence of single-domain proteins. There are many one-word sentences, so to speak, and the stochasticity of protein architecture evolution. I should note that multi-domain proteins are primarily involved in various forms of signal transduction, signal transmission within cells. So the sort of grander hypothesis that I want to propose is that this amount of information gain reflects the minimal complexity to require the informational coherence of a cell to maintain an informationally connected cell, so to speak. Okay, and very briefly, in the last few moments, I would like to show you something that may underlie these mathematical properties of domain and probably bigram frequencies. I will go through this very, very quickly. This is comes from a rather old work, so-called birth-death innovation model of gene family evolution, based on similar to well-known Yule models, well-known to mathematicians that reflect protein duplication, gene duplication, elimination of genes, and influx of gene from the outside, as in horizontal gene transfer. Very straightforward, simple models. There is a bit more complexity. We develop a little hierarchy of these models at different level of complexity, additional terms reflecting different dependencies. This is not so crucial. What is crucial is that these simple models, the small complexification of the simplest form, very faithfully reproduce the universal distribution of gene family size in all cellular life forms that we analyzed. So, power law distribution of domain frequencies is readily explained by a simple stochastic model of evolution without postulate introducing, at least not explicitly, any selection. The question I ask, and I honestly do not know the answer, is there, maybe the linguists in the audience would, is there any equivalent in evolution of natural languages? Or are there fundamental differences between the languages of proteins and natural languages? And finally, since this is the last talk in this wonderful meeting, I would like to use this single slide to just suggest some sort of grand program, if you will. We have talked about architectures of multi-domain, or about grammars of multi-domain architectures of proteins. But in reality, there are structural rules, that is grammars, at all levels of biological organizations. Genetic code that Tsachi discussed here, RNA and protein sequences that he also touched upon, genome architecture, macromolecular complexes, etc., up to the biosphere. So, is there a master universal grammar that might be conserved and discoverable across all these levels? I honestly do not know the answer, and even more demanding question, is there something that approaches a common underlying generative model? And here I close, not before I acknowledge contributors to this specific analysis, primarily my former postdoc, now an associate professor at the University of Alabama in Birmingham, Molay Kumar Basu, students from his laboratory, and two senior scientists in my lab, Yuri Wolf and Georgi Karev. And thank you very much for your attention. I'm happy to take questions.
So we have time for one question because we're running late.
This 3.6 information in a language, it seems to be a lot of it is non-grammar nature. It's just there are correlation between words which are non-grammatical. Let's say Eugene, Kunin are correlated, but not because of grammar, right? So in a language, there is a lot of correlation which are of some other origin, non-grammatical, unless you treat grammar very generally.
Okay. So I guess so that is a comment that there are many correlations in texts that do not reflect grammar, that reflect something else. I suppose semantics, actually. I suppose. Similarly, I think there are different types of correlations in genomes that again reflect, if you will, semantics. And these are epistatic interactions, I believe, between different genes and different parts of the same protein. These, to me, this is distinct from grammar. These are, let us say, synthetic little mutations, whereby mutation A and mutations B in itself are not particularly deleterious. Together, they're little. And there are many such dependencies are pervasive within genes and rather common between genes. So yes, I believe there are different levels of phenomena that define non-grammatical correlations, but I will also claim that these functional rules, what I call grammar, are important in defining biological functions.
Any more questions? Everybody's very... Tzachi, did you want to say something at the end?
Yes, I do.
Want to go first?
Yeah, it makes more sense. Shall we have it? So once again, Tzachi is being very generous in letting me go first. I have to say that this was really Tzachi's brainchild, this kenes, and I was very happy to help in any way that I could, but really it was your insight. And I think I'm wowed by the whole experience. I mean, I think that your insight that biology and language ought to be related because they are part of language has to be part of biology, but in a way that's more nuanced than what is often considered to be the case with language and biology, which usually is restricted to brain structure. I think that there's much more that's that the parallel ought to be developed, and I think that this was a fantastic start. And including art, it makes perfect sense. I mean, this is all part of the. I don't want to use these wishy-washy words like our experience on Earth, but something like that. It's all part, in some sense, it's all part of the same thing, but by inviting such distinguished scientists, I think that we began to see where we can enlighten each other. So I thank you.
So thank you, Wendy, for the kind words. But this really would not have been possible, at least not to this very high level, without Wendy's contributions, which were immense. I wouldn't have dared to do this interdisciplinary meeting without a proper linguist, because of course I'm not. So I thank you for that, and I thank you for inviting colleagues from the linguist arena, which are world class, that definitely facilitated this discussion. So we're finishing now, and you know, I think our heads are really full of new ideas, some of which we understood only very, very partially. It was a very eclectic meeting. You know, it's hard to predict if at all it would be meaningful to any of us, fruitful. Maybe we just had fun for 2 good days. I think creativity comes from very unexpected experiences and exposures and mixes. So this is what we try to do here. So maybe, I don't know, maybe we'll find a gene that is an anagram of another gene or something. Maybe we'll find all sorts of interesting and totally unexpected connections from what we heard here across many different disciplines to what we do. And maybe none of it will happen. It's really impossible to tell. But just towards the end, I want to thank people who helped make this meeting possible. So I don't know if Irit Wexler is here from the Weizmann conference. Ruth Towers from my lab, who worked on this conference forever. Orna Dahan, who is definitely here. She's also my research associate. While I flirt with linguists, etc., she makes sure that the lab is there. So thank you, and had major contributions all along the ways. I admire you guys, and how do you do that? Yeah. And you and all the interpreters and the artists that participated, and I thank all the speakers who agreed to come and share with us their insights. Thank you very much. And I thank foundations at Weizmann that supported all of this. This is without any registration fee. We are very proud to be able to offer such conferences. So this is for Weizmann. And, and now we can enjoy dinner. And I hope all of you would join us to the last bit, which is an art show. It's a play. There's Uti. It's let me explain what it is, because it might not necessarily be familiar to all of you. This is a playback theater show. It's a sub-genre of improvisational theater. It's a theater show that is based on stories from the audience. So you guys would be invited to come to stage one at a time, and we'll do 4 of them, I guess. They will do. The actors are already warming up. It's very interesting, very entertaining. We'll have it in a different building. I hope it will not reduce the number of participants, but we'll walk together. But first, dinner. So enjoy dinner, and hopefully the show too. Thank you.